So good morning and welcome to the fifth annual TIC Academy. The TIC Academy is a product of the TIC IPM Working Group, which since 2013, the TIC IPM Working Group has worked to organize and expand the network of diverse stakeholders working to reduce the risk of exposure to infected ticks by collaborating on IPM-related activities, exchanging knowledge, and sharing resources effectively. The TIC IPM Working Group is run by the IPM Institute of North America, which has been working for over 25 years to increase adoption of integrated pest management and improve sustainability in agriculture and communities through a variety of programs. The organization has two sections, Sustainable Food Group and Sustainable Communities Group. The TIC IPM Working Group is part of the Sustainable Communities Group. The Sustainable Communities Group has three major projects: Green Shield Certified for pest management providers, facilities, and landscapes, the Pest Defense for Healthy Schools program, and the TIC IPM Working Group. If you are interested in learning more about any of these programs, please feel free to reach out to either myself or Josie Dillon, who is also on the call today and will be helping with technical support. The working group does have a website. Is currently in development and receiving some upgrades, but we would love for you to visit it. It is a section of the IPM Institute website, so it's not the old working group website, which is the first one that appears when you Google TIC IPM Working Group. It'll be the second one. But we're excited about some of the changes that we're making, and we'd love for you to go check it out. The working group hosts webinars on the second Wednesday of every month at 1 p.m. Central Time. Webinars are around 1 hour long and cover a wide range of TIC-related topics. If you would like to be added to the working group and receive information about webinars, please send me an email. And if you or someone you know would be interested in presenting at a future webinar, please contact me. My email is on the screen and I think in the chat as well. We also have a YouTube channel. The YouTube channel will host all of the webinars that we have had, that we have recordings of. So previous webinars are on there already, and you can go see the types of presentations that we have and review your favorites and share with your friends. And this is meant to be a resource on these topics. So you can access the YouTube channel by searching TIC IPM Working Group on YouTube or through the link on our website. We do have some continuing education units available for today's workshop. The 2024 TIC Academy has pesticide applicator CEUs available for Michigan, Oregon, Texas, and Washington. And there are vector CEUs available for California. So if you are from any of those states and are attending for the purpose of continuing education, please send me an email with your name, your license number, and your state. And I will make sure that you're on the attendance sheet and all of that fun stuff. You will also need to make sure that you are answering all of the poll questions for today. Which brings me to the format for today. So here you can see our lineup of awesome speakers for today, October 17th. And the way the day is going to work is there are 25-ish minute presentations, which will all include a couple minutes of question and answer at the end. There will be a poll question at the beginning and end of each presentation. Please answer to the best of your ability. Audience participation is highly encouraged, and like I said, there will be a Q&A at the end of each presentation. So if you have questions for our speakers, please type them into the Q&A section, and we will ask your questions to the speakers at the end of the presentation. So if you note on Zoom, we have both a chat and a Q&A. So let's use Q&A for questions for presenters and the chat function for more general questions, comments, that sort of thing. So encouraging you to use both, but as I mentioned before, Josie Dillon is on from the IPM Institute, and she will be helping with technical support, answering your questions, monitoring the chat, that sort of thing. So thank you for your help with that. And if you have questions throughout the day that need to be answered via email, please reach out to her as well. There will be a 30-minute break around 11:30 a.m. Central Time, and then we'll come back at noon with more presentations for you. This session is being recorded, and I will be sending out a recording to everyone who has registered. So if you miss part of the day, don't worry, you'll be able to review it. Yeah, and if you have any questions about any of those things, please feel free to reach out. Finally, we want to thank our sponsors for making the Tick Academy possible. Our primary funding is from the North Central IPM Center. And then we have our gold sponsors, Tickbox Technology Corporation, Project Lyme, US Biologic, and Mainly Ticks. We have our silver sponsor, North Shore Mosquito Abatement District, and our bronze sponsors, Alexis Chesney, MS, NDLAC, and Central Life Sciences. So thank you very much to our sponsors. This would not be possible without their support. If you could go to our website and click on their logos, you can visit their websites. Please check them out and give them your support as well. And lastly, thank you to the North Central IPM Center. The Tick IPM Working Group is supported by the USDA National Institute of Food and Agriculture Crop Protection and Pest Management Program through the North Central IPM Center. And we are currently in the process of applying for future grant support. If you want to support the work we do, and you like what you saw here today, please consider writing us a letter of support. It will go a long way towards securing future funding. And for more information on letters of support, please contact either myself or Josie Dillon, and we will send you our template and some instructions and that sort of thing. So thank you all for being here. I'm very excited about the speakers we have today, and we're going to transition to our first presentation in just a second. Josie, can you please launch the first poll question? And Dr. Chinnici, can you please share your screen? So while you all answer that question, I'm excited to introduce Dr. Nicole Chinnici, who is the director of the Dr. Jane Huffman Wildlife Genetics Institute at East Stroudsburg University, where she has focused her research on ticks and the pathogens they transmit. Dr. Chinnici pursued advanced studies in epidemiology, where she delved deeper into the complex ecology of ticks and the pathogens they carry. Her work spans fields such as microbiology, epidemiology, and other molecular genetics, with an emphasis on tackling this global health challenge. Under Dr. Chinnici's directive, the Tick Research Laboratory was established in 2018 to evaluate the dynamics of tick populations and the factors influencing the spread of tick-borne pathogens. Findings of the research will contribute to the development of innovative strategies for disease surveillance, risk assessment, and vector control, enhancing our ability to combat emerging threats beyond her scholarly pursuits. Dr. Chinnici is deeply engaged in outreach and education efforts aimed at raising awareness about tick-borne illnesses and promoting preventative measures. She's a devoted advocate for public health initiatives and policy measures aimed at mitigating the impact of these diseases on both human and animal populations. Dr. Chinnici, thank you for joining us this morning. The floor is all yours.
All right, thank you. Good morning, everyone. We'll go ahead and jump in, and I hope I could stick to the time frame that we have here. So I'm Dr. Nicole Chinnici. I work at the Pennsylvania Tick Research Lab at East Stroudsburg University. We'll talk a little bit about what we do and some of our research that we're diving into. So Tick Research Lab is housed at East Stroudsburg University under the Dr. Jane Huffman Wildlife Genetics Institute. The Wildlife Institute does a lot of various molecular research. So it's a fully equipped research laboratory, about 8,000 square feet, with state-of-the-art clinical equipment to do molecular tick testing, as well as research on small mammals and other wildlife species. We have ongoing research, and our main focus is focusing on ticks and tick-borne pathogens, and we've been focusing on research since 1998. So we have a really big archival database of samples and results from that time span. And we started our statewide surveillance programs in 2018, and we have both a passive and active ongoing surveillance project that I'm going to talk a little bit about. So we'll talk first about the passive surveillance that we're doing in the state of Pennsylvania. So we have a website that residents can go on and submit their tick in for testing. And with if they're a PA resident with a Pennsylvania address, they qualify for free tick testing. Ticks are then mailed and dropped off to us, and we do qPCR testing of 5 different tick-borne pathogens on those ticks. The turnaround time is about 2 to 3 days, and the purpose of that is to collect a lot of important information as well as to give the individuals information about what they may have been exposed to. So what we've been doing with this passive data is tracking tick-borne diseases in Pennsylvania, what diseases we're seeing in the tick populations, what maybe changes have been occurring over the last several years. We're looking at distribution of ticks and exposure to different ticks. So are there any tick species emerging in our state? Is it mainly just the black-legged tick that individuals are exposed to, and what does that look like? We're looking at demographics of tick bites, and I'll talk about how we're using that information to drive some public health alerts and some trying to change some behaviors and how individuals are interacting with their environments. And then on the flip side, it does give individuals an identify whether or not they've been exposed to a tick-borne illness. It identifies risks, so looking at how engorged that tick is, and then they can use that information with their physicians to better understand potential symptoms that may arise. So what's included in our free tick testing? So Lyme disease for any ticks that's submitted, and then depending if it's a black-legged tick or a non-black-legged tick, we test for several other tick-borne illnesses: anaplasmosis with its variants, relapsing fevers, babesiosis, Powassan virus, Rocky Mountain spotted fever, Ehrlichia. The list is essentially selected based off of the most common pathogens that we were seeing in the environment, but it has shuffled a little bit over the years based off what we're testing for. So a little bit about some of the results that we're seeing. So we tested, this is 2023 data, based off the black-legged ticks. Total ticks that were tested in our state was a little over 18,000. When we focus in on the black-legged ticks, what we see, the adult black-legged ticks are the majority of the ticks that we have for submissions, about 8,600. And 58.6% of them test positive for at least one of the tick-borne pathogens that we test for. So that could be Lyme disease, anaplasmosis, babesiosis, Powassan virus. And the state co-infection rate is around 8.39% with a range from 6.5 to 10.6. So quite a number of ticks, and especially the black-legged ticks, come in infected as well as co-infected. Our nymph black-legged ticks, about a third of them test positive for a single tick-borne pathogen. And all of us know that the nymph ticks are considered the most dangerous because of their size, and most of us are very good at seeing them or identifying them until they've been on for quite some time. And their co-infection rate's just shy of 4% in the state of Pennsylvania, with some varying rates. And I put the ranges on there because I think it's really important when I'll highlight some of the results that we're seeing across the state of where these ranges are occurring and what might be happening. When we look at how these co-infection rates are appearing in the environment, if they are at random or if they have any positive influence on each other, we see that most of the time this is looking at the heat map, this is adults, and this is the nymph ticks that are tested. None of them have a negative correlation. Some of them are at random, but most of them have a positive correlation with they're occurring more frequently in their environments than they would at their individual pathogen rate. And we see this really, really strongly with babesiosis. So babesiosis co-infections, when we look specifically at them, where they're occurring and how frequently they're occurring, you can see this is a couple of years of our data, all of our positive ticks. So just shy of 800 positive Babesia microti positive ticks that were tested. Of those positive ticks, only 25% of them were single infections, whereas almost 61% of them were co-infected with Lyme disease. So it shows that positive synergistic relationship between babesiosis and Lyme disease, and the ability for babesiosis to thrive a little bit better in the environment when Lyme disease is present. Some of these other pathogens, human anaplasmosis, Borrelia miyamotoi, deer anaplasmosis, and deer tick virus are less common, but with Lyme disease, the co-infections were much higher. And we see this, these black dots are where all of these Babesia co-infections have occurred, and you'll see this sort of concentration gradient where more of them are occurring on the eastern portion of the state and less on the western portion of Pennsylvania. And when we look at our actual ticks that we tested positive from 2023 data, we see the same thing in this eastern, higher proportions of infections in the eastern portion of the state compared to the western portion of the state. So these are the total number of ticks, and you'll see relatively based off of the regions, there's pretty similar population or sample size with the ticks are tested. Southwest tends to have a lot more just because of the Allegheny and Pittsburgh region and more the highest human population. But you'll see the total number of ticks that test positive, blue is adults, and then red is the nymphs. You'll see higher rates in the eastern portion of the state, and then as you move central to the west, those rates drop down. So we're seeing something that's going on with babesiosis and these infection rates and how this is spreading. So the essentially what I'm trying to show here is this passive surveillance study is allowing us to gather information about what's going on with these pathogens and how these pathogens may continue to emerge and affect the public health as well as the residents of this state. And how can we use that to sort of take some measures and what we can do. So we do a lot of public health outreach with this information to help our residents better understand how to recreate in our state safely. Some of those things is taking our data and being able to provide them with some more specific tips of what might be going on. So we send out alerts. We're very common on social media, trying to create as much informative information with our data as possible to help drive some behaviors as well as awareness surrounding. So these are just some examples of that data being converted into some social graphics that we share out to our residents. One of which just went out giving that peak season alert for the adult ticks that are emerging in our state currently, exposing individuals to those. We also take the data to get a better understanding of exposure and where ticks like to attach. This helps us better understand and guide individuals with checking for ticks and how we can remove them a little quicker. So when we do ask individuals to submit a tick into us, they take a short, brief survey, and then we can use that information to drive some education. So we do ask where the tick was removed. And this is specifically taking any adult black-legged deer tick submissions and quantifying and summarizing what those areas of reported attachment were. And you'll see that most of them tend to, and this specifically for the adult, will attach to the hairline or behind the ears. We also dive a little bit deeper into that data and looking at what else we can use from this data to extrapolate and drive some more information. We've identified that the greatest risk of exposure to ticks in our state has specifically been in the backyard, and a lot of that has been conducting activities such as yard work, playing with the kids, playing with the dogs in the backyard. So that was more than 50% of our exposures have occurred in the own backyard, which has been different than some of the mindsets of our residents prior to. They always thought tick exposures would be when they went to the parks, when they went out hiking and things like that. They weren't realizing their backyard was their biggest risk. So trying to drive home to really practice tick prevention whenever you go outside. We did have some data that showed that the area where the tick tended to attach was based off of the activity. So that helps us to better drive education. So if you're out gardening and your hands are in the leaf litter, or you're doing yard work and you're raking the leaves, your risk of attachment will occur usually on the upper body. Whereas if you're out walking around, hiking, things like that, the ticks will tend to attach a little bit lower on the body. We have age distribution of our ticks here in this map. Everyone's probably seen the map of Lyme disease cases, and you'll see that the exposure to ticks is also overlaps pretty much the number of cases that we see in tick-borne illnesses, where the majority occur in our children under 10 years of age. You get this little peak in our 30 to 44-year-old, and it drops back off, and then you get this other peak over the ages of 55. We look at the average length of time a tick is attached. How good are we at identifying a tick and removing it before it's been attached too long to be a risk for transmitting illnesses? Adult ticks were pretty good. We're getting them removed within 7 hours before they become engorged with blood. Nip ticks were not so good. This is aligns directly and correlates with our development process that the nymph ticks are the most dangerous. It does take on average 18 hours before someone identifies a nymph tick attached to them. And then moving into our other portion of our data is really looking at the active surveillance. So we have understanding of our exposure with our passive surveillance on this side of the equation, and now we're on this side of the equation, really just looking at the hazard associated with tick-borne illnesses in Pennsylvania, and what makes Pennsylvania a hotspot for these tick-borne illnesses. So we've launched an active surveillance project. We're in year 2 of that. We've just finished our baseline and we're still summarizing our information. And a lot of what we're quantifying in this is understanding the density of ticks and what environmental factors may influence that density of ticks, the density of small mammals in our areas and in our environments, occupancy of our large mammals, what's coming through the area, and how that may play a role in changes in infections as well as tick densities. Densities of the tick-borne illnesses in general, and we're using some vegetation composition at each one of our sites to see if this influences what we're seeing, because we do see site-to-site variation in both densities of ticks, small mammals, and infection rates. So what could be the factors that really drive these infection rates, and then how can we use that in the future to really drive some of these tick mitigation strategies and how we select that. So our study area, we're on 2 parts of the state. I highlighted that map where it showed the babesiosis and how some of these infections are more concentrated on the eastern portion of the state versus the west. So it was reasoning for us on selecting 2 counties on the eastern portion of the state and 2 counties on the western portion of the state to kind of quantify some of those variations that we might be seeing. And our goal is to secure some more funding so we can at least be in all 6 regions of Pennsylvania. But for right now, we have 36 sites, 9 in each of our 4 counties. In the Northeast, it's right here is Pike and Monroe County. And then in the Northwest, we have Mercer and Crawford County that line up and butt up against Ohio. And we butt up against New York and New Jersey on the eastern portion. Our sites are varied. We have schools, parks, residential communities. Really, the goal is just to kind of get a nice diversity of interfaces that we may encounter ticks based off of individuals' normal activities with the environment. So what do we normally do on our day-to-day activities, and then take it into a combination of all those so we get real good picture and understanding of what's happening. And then at each site, we have, they're pretty big. We run pretty much, we try to run along the trail edges if it's a trail, or the forest edge. So this is an example of a park here where there's this trail that goes around the entirety of the park that we run our plots on. We try to do 11 continuous plots, but you'll see here there's some breakage in that. It's just based off the landscape and what the site has to offer. They're about a half mile. Some of them are linear, some of them can be circular. At each one of these plots, they're 750 meters square, so they run 750 meters linear and then 10 meters into the wooded edge. We have these subplots where we do vegetation analysis to get an understanding of the variations at each one of these. And we do small mammal trapping every 10 meters on the inner portion of these plots when we do so. When we look at our environmental variables that we're collecting, we're looking at canopy coverage, how much sunlight these areas are getting, leaf litter depth to get an association of what the leaf litter may have in terms of creating microclimates. We're also collecting soil moisture data, vegetations. We're looking at grasses, forbs, bare ground versus rock, what types of plants are in the area, elevation, slope, various different factors that may influence changes in density of ticks as well as the small mammals. And we're collecting temperature and relative humidity on a continuous, continuous basis at each site. We're doing tick collections, so we do target tick collections for our density of ticks. So we look at questing of ticks during 2 times during the adult season and 2 times during the nymph season, and then we quantify that to get density. We're also collecting ticks off the small mammals as we capture them to get an understanding of what ticks are feeding on them, and that'll also help us look at the number of ticks feeding before we do any treatments versus the number of ticks feeding after we do treatments. Small mammal densities, we're doing a 3 to 4 night consecutive trapping where we're setting out, it's site dependent, 73 to 91 traps a site, checking them every morning, collecting ear blood and ticks. And any new individuals that we handle, we're giving them subcutaneous RFID pit tags. This way we can do some modeling for densities and mark recapture rates. We're doing camera trapping. We put 2 cameras at each site, 15-day intervals, scented and unscented, looking at large mammal occupancy. And then just to dive into a little bit of our results that we're finding in terms of ticks, and we're still in the process because we collected a lot of samples our first year for baseline, and we're about halfway through our second year, and we're still summarizing some of this data to get an understanding of what's going on out there. So in terms of ticks via tick dragging, we got just shy of 20,000 ticks across all 4 of our counties. The diversity wasn't very high. Mainly, we were capturing the Ixodes scapularis, adult and nymphs, and larvals. Some dog ticks. Interesting here with dog ticks is we do get a lot of dog ticks in Northeast Pennsylvania, but very few in Northwest Pennsylvania. And then we have some other few ticks that we've collected, like the Gulf Coast tick and Lone Star ticks randomly in the Northeast, but have not seen any in the Northwest. Diversity on the small mammals is different than the diversity we're seeing in drags. We've identified some brown dog ticks as well as some Ixodes angustus on our small mammals, but these ticks weren't found via our drags. Density of ticks, this mimics the tick season and our understanding of what happens on the cycle of the adult ticks. So the adult ticks are less dense in the summer, more dense in the fall, where the nymph ticks are more dense in the summer and less dense in the fall. But one thing here to note, when we're looking at some of these geographical differences in between our site areas, our Northeast sites, our density of ticks is much lower than our Northwest sites. And that's in the adults. The nymphs, they're slightly higher, but not too much different between the 2 regions. So just some interesting things there that we were starting to see. Disease data is just currently summarized for our ticks collected in the Northeast. We haven't finished up our Northwest. It's important to note here for our adult ticks that we see almost 50% of them were infected with Lyme disease in general, but you'll see some differences in county level, which is important to note that county to county, even site to site, there's some differences in these infection rates. And we did identify all the pathogens that we're testing for. So this is Lyme disease, anaplasmosis, Babesia microti, Borrelia miyamotoi, and deer tick virus. And then our co-infection rate, again, some pretty big differences in a co-infection rate between counties. And then the nymphs, again, identified all the tick-borne illnesses that we were testing for. We did not find any deer tick virus in Pike County for the ticks that we have already analyzed within the nymphs. And then you'll see our co-infection rate. When we look at our small mammal captures based off so the Paramyscus leucopus is the white-footed mouse, and looking at their population sizes across each one of our studies. Green's going to be higher densities and red's going to be lower densities. And you'll see here's the eastern portion of our state. We have much higher mice densities than we do on the western portion of our state, which again goes back to seeing some of those infection rates may be directly influenced by our population of small mammals. Babesia microti infections in our small mammals. This is the eastern portion of our state. Sample size is the larger the circle, the greater the number of individuals sampled. And red is higher infection rates and yellow is lower infection rates. And you'll see we have Babesia microti across all of our sites except for 1. in Northeast Pennsylvania was detected, and we only had one site in Northwest Pennsylvania with a single animal tested positive for Babesia microti. So again, showing that eastern to western potential emergence of this tick-borne illness. Anaplasmosis, we did see more anaplasmosis infections in Northeast than we did in Northwest, but some more infections in comparison to the Babesia microti. So we really are starting to dive into what might be driving some of these environmental influences. And again, a lot of this is preliminary data because we're still summarizing all of the information we have regarding these illnesses. So when we look at variables of what's affecting our small mammal population, we find that relative humidity, elevation, and infection of Babesia microti influence larger populations of the small mammals. And then when we take all of our regions in comparison, we see that Babesia microti infection is a big driver for increase in small mammal populations. So it's very interesting. And we're trying to dig a little bit deeper into what might be the reason for that. So looking at what environmental influences tick densities, when we include tick densities on our small mammal populations, we are seeing that relative humidity has a negative effect, and the small mammal population size has a positive effect, as well as the tick density. And the population size, again, has a strong influence. So the larger the population size of those small mammals, the positive effect it has on whether or not there would be a Babesia microti infection. And then looking at the same thing with Anaplasma phaglocytophilum, temperature played a role when we looked just at our Northeast sites, but there was no influences when we looked at our total. So what we're doing now with our active surveillance project, we are still summarizing our year 1 of data, but what we're diving into is some mitigation strategies to see how we can ultimately reduce the tick population in our state, taking into account all these different variables. So we're looking at bioinsecticides, and we did start applications this past summer. And we're looking at the Lolligard M52, which is a fungus spray targeting ticks. And then we're looking at using, in collaboration with US Biologics, their bait station technology with anti-tick and Lyme disease bait vaccines, and how that will influence tick attachment and tick-borne pathogens within the small mammal populations. We're doing combination sites and control sites to evaluate this over several years to see if we can come up with some strategies to reduce our burden of tick-borne illnesses in our state. So these are just some pictures. We have a mouse right here in our station consuming a bait. We also get some other friendly visitors interested in what's going on. So future directions, just to wrap up quickly, I know I'm going over time. We're continuing to process our genetics data, really understanding what's going on with the pathogens, accounting for some new variables like atmospheric pressure, soil moisture, and snow death over the winter, and how that might influence lagged environmental effects with some acorn crop, accounting for differences in seasonality and recruitment and loss rates with our small mammals. We're working on potentially adding on some more sites and counties, and then working on improving our occupancy analysis with some simulations and trying to figure out how those large mammals could be playing a role in what's going on in the environment. And then we'll be starting to evaluate our tick mitigation strategies next year after we've done a year of applications. So thank all of our funders. We're funded through Pennsylvania Department of Health, Department of Education. Ticklab.org is our website, and we're very active on social media. You can follow us at PA Tick Lab. So thank you, and if anybody has any questions.
Thank you, Dr. Chinnici. That was very good, especially since we started late. So I'm sorry about that, but we're still going to try to get a couple questions in. The first couple sort of go together. There's a question about other small mammals. What other small mammals are you sampling, and are you considering birds in your surveillance?
Yeah, so we're just doing Sherman trap sampling. So we do get non-target species. Peromyscus leucopus is our most common small mammal that we capture. It's probably about 99% of our captures. We do have some sites that have Peromyscus maniculatus still. So we do have those. We do have a handful of shrews that are also captured. That we've been evaluating and have some other studies going on with them, but the main is the small mammals in terms of the mice. We're not doing any bird sampling at the moment.
Thank you. Josie, can you please launch the post question while we answer another question? With the average length of attachment time, was this self-reported, or did you guesstimate based on the engorgement level?
Yeah, so we do it, we use a microscope and we're looking at engorgement of the shield to body ratio to get an estimated level. I think kind of similar, tick exposure risk in the backyard, is that confirmed / was it self-reported data? It is self-reported data, so we do ask where individuals, what they were doing and where they think they've been exposed. So it is self-reported data for that one.
And the last question, just try to keep us close to the time. Have the protocols for tick pathogen testing been calibrated with CDC pathogen testing or DEP pathogen testing?
Yeah, so we follow the CDC guidelines for surveillance in all of our pathogen testing.
Perfect. Well, Dr. Chinnici, thank you very much for your presentation. Thank you, everyone who's here for your questions. We are going to transition to our next presentation in just a second here. Second, get everything going. There we go. Morning, Mark and John. Are you?
Good
Oh,
morning.
screen check. Perfect.
All right. Good morning.
Good morning. Thank you for joining us. Let me give you a little introduction here. For our next presentation, we have Doctors John Carlson and Mark Janowiecki. Am I saying that correctly?
Yeah. Yep.
Dr. Mark Janowiecki is a research entomologist who has worked with the city of New Orleans mosquito, termite, and rodent control board since 2020. He has always had an interest in insects and studied termites throughout his degrees, applying molecular tools for his species and population differentiation. He currently leads the termite division for the city, which protects city facilities and historic buildings and trees. Mark also manages the molecular laboratory, which conducts pathogen surveillance in mosquitoes, rodents, and rodent ectoparasites, genetic resistance, screening of mosquitoes and rodents, and population genetic analysis of termites. And Dr. John Carlson received his medical degree from Tulane University School of Medicine in 2004. He's board certified by the American Board of Allergy and Immunology. Dr. Carlson performs pediatric allergy and immunology performs pediatric allergy and immunology and sees patients at the Ochsner Center for Primary Care and Wellness. Thank you both very much for presenting for us today. You can begin whenever you would like.
Well, great. Thank you very much. So this is really fun for me. So I am a physician and my specialty is the immune reactions to arthropod antigens. So I study asthmatic responses to inhaled cockroach proteins, and then I do a lot of venom allergy research and clinical care for all of the people who get stung down here and have anaphylactic reactions. And then over the course of my career, I've watched the development of a new diagnostic entity, this alpha-gal syndrome. We knew that ticks were bad, as you've heard from the previous speakers, and from your own experiences. Clearly, there are a lot of pathogens that these ticks transmit. But going on to the next slide, Mark, do you have control? Yeah, thanks. We started noticing a really bizarre pattern of cross-sensitization. So cross-sensitization does happen, but it happens when there is a clear second mistake of the immune system. The first mistake of the immune system is any allergy. So the part of your immune system that does allergies is supposed to be fighting off the ticks and other parasites. But what happens instead is it might start targeting birch pollen as one example, or cat hair, or something else that your immune system mistakes as a parasite and begins to make allergic antibodies against that substance. Rarely, the particular part of the allergen that it targets is very similar to an epitope on another allergen. So big examples that have been well described are that people who are allergic to their cats, so you get sneezing and eye itching. If you eat pork, you can have an anaphylactic reaction because of a shared epitope on cat albumin, if that happens to be the epitope that you're allergic to. Similarly, much more common is birch pollen allergy, resulting in a local allergic reaction when you eat apples because of this BETV1 protein that's very similar between the proteins in apple peels and birch pollen. But this new one, I was around long enough to actually watch this be discovered, that people would develop a tick allergy, a local allergic reaction, which is an appropriate immune response to ticks, but subsequently would start developing allergic reactions when they would eat meat. Next slide. I was so perplexed by this and followed it that I was invited to write sort of an op-ed in one of the big allergy journals to describe how confounding this particular cross-sensitization is. So in this chart from the paper, you can see what is typical for allergic reactions when you have a food allergy, and then what is different about alpha-gal syndrome. On the bottom line there is probably one of the most frustrating things, is that the actual reaction is highly inconsistent. There are a lot of cofactors and other variables that we don't understand that can change how long it takes to have a reaction, and what symptoms will be involved. So for example, if you have a primary peanut allergy, every time you eat a peanut, your reaction will probably be similar with each encounter of the peanut. That's not true for alpha-gal. One reaction might just be an upset stomach. Another one could be full anaphylactic shock. Next slide. Also confounding, that is very unlike other types of food allergy, is that the reaction does tend to fade over a couple of years if you avoid the tick bites, the primary sensitizer. But bizarrely, other things can boost your reactions to the meat allergy. So tick bites certainly can do it, but beyond that, flea bites, chigger bites, and the Hymenoptera sting, so like a paper wasp pictured here. There is no shared epitope between the venom in these Hymenoptera, the social wasps and honeybees that would sting and can independently cause allergic reactions. But there should be no shared epitopes, no shared allergens with this alpha-gal syndrome. And so this is really, really perplexing. There are so many unanswered questions with regard to alpha-gal syndrome. Next slide. One of the frustrating things in managing patients with alpha-gal syndrome are the very limited tools that we have for the diagnosis. So if you're allergic to peanuts, I could do a skin test or a blood test to determine whether or not you have allergic antibodies. For alpha-gal, there's no skin test available. You can't buy the extracts to perform the test. The blood tests are available, but they're not reliable. Many people, most people who you can detect allergic antibodies directed against this alpha-gal, this sugar epitope that's cross-reactive between the tick saliva and the meat. Most of the people who have those allergic antibodies actually have no symptoms whatsoever when they eat meat. So very unreliable testing. The more reliable testing that we have are the ingestion challenge. And the classic is a high exposure ingestion challenge. So for example, you say, come on into clinic and we'll feed you some sausage or some bacon. So that is reliable if you do a high exposure ingestion, but it's dangerous. You trigger anaphylactic reactions. And so we have to do those in our specialty clinics that are designed to manage anaphylactic reactions within the clinic. So I run a high-risk allergy clinic, so that's what we do. Other places will do low exposure ingestions. So there is some alpha-gal in ice cream. And so the reactions, if you have it to ice cream, tend to be pretty mild. So it's safe. But just because you can eat the ice cream doesn't mean that you would be able to tolerate bacon, for example. So it is unreliable but safe. Next slide. In terms of the inconsistency, there is very small amounts of alpha-gal in things such as the gel caps that are common for medications. Most patients who have alpha-gal syndrome are able to take the medicines in gel caps, but not all. Some will have anaphylactic reactions just from taking some naproxen, depicted here. More scary are these plasma expanders that have a large amount of alpha-gal. This is if you get in a car crash and you've lost a lot of blood and they need to make sure that you can have some fluid in your blood vessels to maintain your blood pressure, you'll get infused with these plasma expanders. And unfortunately, really horrible alpha-gal reactions can happen. Because this is a relatively new and quirky diagnosis, most physicians aren't familiar with alpha-gal syndrome. And most allergists who are familiar with alpha-gal syndrome, because of the complexity and the danger involved, don't feel comfortable managing the patients. And certainly most will not feel comfortable for doing the high dose ingestion challenges. Because it's a rare disease and it manifests in so many different ways, it is incredibly difficult to do research on this. There's really one center in North Carolina that does most of the clinical research on alpha-gal, although there are sites in other countries where more and more research is being done. So hopefully we will begin making some progress. Next slide. So this is where I'm going to hand it over to Mark, who is an amazing person, and he's going to describe his experience with alpha-gal syndrome. He is unusual in that he, as an entomologist, has a very firm understanding of exactly what's going on. So let me pass it over to you, Mark.
Awesome. Yeah, thank you. And thanks for the great introduction to alpha-gal and some of its strangeness. So yeah, I'm going to kind of veer from the more scientific into more the personal story of my experience with alpha-gal. So to go through this, I want to share a little bit about my background, kind of how I picked it up and how I discovered I had it. So I'm originally from a small town in Northwest Ohio in Findlay. From there, I went to study entomology at Ohio State, graduated in 2012. A little side trip down to Disney World to do a pest control in the greenhouse internship in the land in Epcot before going to Arkansas. And so this is the more important part of the story. I did my master's degree at the University of Arkansas from 2013 to 2015. And this time we would spend a lot of time in the woods. This is me out collecting. This could be the day I had many tick bites every day that I was out. So this could be the day I got alpha-gal. I don't have it pinpointed down that specifically. After my master's, I went on to do my PhD at Texas A&M University, kind of out of the range of alpha-gal syndrome, which we'll talk about, that kind of complicating my story a little bit. And then now, as was mentioned in the introduction, I work for the City of New Orleans Mosquito Termite Rodent Control Board. I'm going to focus in the story on just this part here where I was in Arkansas and Texas A&M, as this is kind of the main part for the tick side of the story. So I mentioned when I was at the University of Arkansas in that area, there were many ticks. I got many tick bites. As a consequence, I just kept a pair of forceps in my shower. So every time I came back from being out in the woods, I would have a tick to pull off. So it was inevitable. I likely contracted alpha-gal just knowing the range and where this is expected. This was likely the time when I picked up alpha-gal syndrome, although as I mentioned, I don't know the exact date or time. From there, I went to Texas A&M and started having symptoms that first fall after leaving in 2015. But many of these, I didn't really know what had happened. So I knew that something had happened in the night. I had an upset stomach or whatnot, but I didn't know if it was food poisoning or, kind of had a foggy memory of, feeling sick in the night, but feeling fine the next morning. My first real documented reaction came the next fall in October of 2016. And this I want to get into because usually as soon as someone knows I have alpha-gal, the first question is, what's the reaction like? So my reaction starts off with itchy hives, especially in the joints between fingers, hands, ankles, knees, back of the ears. You get a lot of redness and swelling, particularly in the hands. It gets to the point where my fingers can't close into a fist anymore because they're so swollen. I can't really use my fingers. It comes with a drop in blood pressure, so I get lightheaded, fainting, can't even really make it over to the bathroom and back without having to lay down on the ground. I get kind of a wheezy cough with a deep breath, and then an upset stomach with diarrhea. And this is an example of my forearm showing a lot of those hives during a reaction. And so the thing that I also want to stress is that, I was educated in entomology, but didn't really know the medical side of things. And I didn't realize, I thought anaphylactic was the characteristic in a movie where you eat a shrimp and your lips swell and your throat swells and you can't breathe. But anaphylaxis is just multiple systems responding. So it doesn't even have to be that breathing issue. It's still a major concern here. I also want to highlight, as Dr. Carlson mentioned, the variability of reactions. So my reaction is not universal. This is how it goes for me. But if someone says they have alpha-gal and they're, have a different set of triggers or symptoms, this is true. They're not making this up. Some people do get fume reactions where if, in a shared break room down the hall at work, someone heats up a hamburger, they could have a reaction just from the fumes of that going. I've never had an issue with this. Same with dairy. I've never had an issue with any dairy products or any mammalian products. So the gel capsules and things I tried to avoid, but I certainly didn't perfectly avoid and never had a reaction that couldn't be traced to meat. They're also very sporadic and consistently inconsistent, as Dr. Carlson mentioned. So it's difficult to really pinpoint them. I would have a reaction to a food one day, eat the leftovers a few days later and not have a reaction. So the scientist in me had a hard time really understanding what was going on. So as time went on, I mentioned my first documented reaction was in October 2016. After that time, I had, including that, I had at least six reactions to different things, beef or pork ribs or different products. One of these happened to be before a very big exam in my PhD process. And so it was the night before. So the next day I could barely type at the keyboard. My hands were swollen. And I had a hard time completing that test. But so that's when I first started to have, an impact on my life where I wanted to go see an allergist and try to figure out what's going on. You can also see the frequency really increases. So it goes from 2016 to 2017, almost a little over a year into 2018. And then it really picked up and was in June, September, October, and another one in October. So I was then diagnosed in 2018 when I went into my allergist in Texas because he had never seen it. He was very skeptical, saying, yeah, we'll test you for that, but I think it's probably something else, and tested for a lot of different things, which all came up negative, and alpha-gal came up as positive. And I remember after being diagnosed, I went to the grocery store probably on the way home from the doctor's appointment and was picking something up to make for dinner. And just as I was walking around, could kind of feel the depression of, these are all the things I can't eat anymore, and remembering more and more things that are, are mammal. And this is another thing I can't eat. I also, shortly after being diagnosed, went to a restaurant that I liked called Piada. That was kind of like an Italian version of Chipotle. Usually I would go and get a steak piada as the primary protein in there and get some bacon added as well. And as I was ordering, I remembered not to add the bacon, but I remembered not to order the steak. I got the chicken instead, but I completely forgot and put the pancetta, the bacon in as well. And so I got home and couldn't eat this. And, just out of habit, had usually had my order. I knew what I always ordered. So it was tricky. And as I got this, there weren't a lot of good materials out there. So it's hard to really find what to do. And so my best resource at the time was a Facebook group for alpha-gal with a lot of members. In this, people will post and say, this is my, my test result came back as this. My doctor doesn't really know what's going on. What does this mean? Or, I had this flu shot this year and I had no reaction. So it's, it probably doesn't contain mammal ingredients in this vaccine. But it was hard because my doctor didn't know what this was or what to do, and it was very difficult to find information. And now there's a really great website, alphagalinformation.org. This has a lot of the material that I spent time kind of discovering on my own, or searching through different means, all together in one place. So this is a really great resource, really great for patients, providers, or even just people who are interested. alphagalinformation.org really summarizes a lot of the things that took me a long time to discover on my own. But some of the things that I found were alternative foods. So we can't eat mammals, but you can eat all the different poultry. So things like ostrich and emu are a really good substitute. I've really begun to enjoy duck as a food source. It's much more like a red meat and really good. There's also turkey options of many things like turkey pepperoni, turkey sausage links. But even these, you have to be very careful because many of the turkey sausages are made for dietary, avoidance of pork, not allergy avoidance of pork. So they'll still have a pork casing around the outside, or the pork intestine that they use to case the sausage. So you really have to look close, and these things aren't labeled. So you can't look at the back and see if it contains pork. It'll have all sorts of different names that they're calling that pork intestine lining, but they don't say it in those words usually. So it's difficult. And there's plenty of other options, like the Impossible Whopper came out. And this, the first time I got it, I was a little concerned because it tasted just like a normal burger, and I was worried they gave me the wrong one. So there's lots of substitutes out there and ways around it, and other things you can still eat. This is an example at one of our work crawfish boils down here in New Orleans, and they were nice enough to make the first batch without sausage in the pot, so I was able to eat it. So I can still have all the seafood I want, all the poultry, chicken, turkey, duck, fish, vegetables and grains, all sorts of things are still available. There's another interesting case where there's gal-safe pigs. So these gal-safe pigs are a genetically modified pig that was initially produced for different transplants into humans to avoid the rejection of these different blood vessels or different pieces that they're taking out of the pig. And they realized they had all this meat that also had the alpha-gal sugar removed from it, and they got it approved by the FDA and are sending it out. It's still not commercially available to buy. They're just sending out kits to people who have alpha-gal of some of this meat, but they send out pork chops and ground pork and bacon, all the different parts of the pig that don't contain this sugar. So hopefully that'll eventually make it onto the market and see if this is available as an alternative. So Dr. Carlson talked about the food allergies of eating sausage or bacon and coming in. And so I participated with those once I got here and was able to work with Dr. Carlson, who had a lot more experience with alpha-gal and was a really good resource for all of this information. So as I mentioned, I had my first symptoms in 2015, diagnosed in 2018. That next summer, I couldn't resist and went and got a steak just to make sure I still had it. And I did. That was actually the hives picture from earlier. But then a couple years later, once I moved into New Orleans, I scheduled a food taste test in August of 2021. And this was the food I missed most. So, you can get a pretty good fake burger, but you can't get a fake steak. So it's hard. So I had a steak, I had corned beef, some pepperoni, lots of bacon, and lots of sausage. So this was the picture of my breakfast that day before I then drove myself in. So unfortunately, with this one, I failed. I began having stomach issues and hives within the first couple of hours, and took epinephrine and was able to have that reaction pass. A couple years later, and just about a year ago now, in October 2023, I found these alternatives, the emu steaks and different things for beef, but I really wanted some bacon, because having, a fatty piece of bacon is not replicatable with turkey bacon. So I went in, I made as much bacon and ate as much as I could, and then went in and ended up eating about 1.5 pounds of bacon. And again, had hived and failed. So I was curious about desensitization and talked to Dr. Carlson, because I noticed sometimes I had some accidental consumption and didn't have a reaction, whether I just didn't read the menu close or it wasn't really mentioned in the menu. There would sometimes be some meat, and I would have, a couple pieces of meat and see what happened. After the taste test last year, I began eating 1/2 piece of bacon every day to try to see if I could kind of condition myself back into this. I increased by 1/2 piece every about 2 months. And by the end, I was having 2 slices of bacon every day. Then after 8 months, this summer, I went to lunch at the Brazilian Steakhouse downtown here in New Orleans, and then went straight over to the doctor's office for my food allergy challenge. So this time I passed. So I went in, in this last June, ate as much of the beef and everything as I could there, and then went in. My stomach was upset just probably from all the meat, but I didn't have any hives, didn't have any anaphylaxis. So from there on, now I've been kind of cured by bacon. And so I've been back to eating meat. Luckily, we went traveling this summer into Poland and had lots of different meat options there. That was really, really good. So I'm back to regularly eating meat and hopefully have desensitized away and have moved past it. But this was a part of my life for about 6 or 7 years. I want to wrap up with some kind of alpha-gal frustrations as a patient. And so these are just kind of some of the experiences that I've had. And some of this is influenced by getting alpha-gal and then leaving the region where it's common, where some of these might not be the case as much in other areas. So the first example, I went here in New Orleans after I moved here to get a flu shot. And I went to a drugstore and saw a nurse practitioner. And I asked, are there any mammalian ingredients in this vaccination? And she countered by saying, are you against animal testing? And I then had to explain that doesn't relate at all. This is not what I'm talking about. I have a condition that she had never heard of and didn't understand and had to look through the ingredients to see. So it's just, even in the medical field, alpha-gal is not known and is still a little bit confusing. There's also been a lot of restaurants not really taking the allergy seriously. There's a lot of people have allergies or people have food preferences, and that kind of gets mixed together. And in the end, it's just not taken as seriously. Where I ordered food and said I can't have mammal or this or that, and tried to really explain what a mammal is in case they're not sure. And the food came out, and there are little pieces of something in it. And I asked what it is, and they said, "Oh yeah, there's bacon, but it's just for flavoring, so it's okay." And that still is an issue. And then the obscurity just in general takes a lot of explanation. Mammal products or mammalian products is not really understood. A lot of people don't know exactly what a mammal is or know that category. So you really have to list out everything that is a mammal and go through all the details. And this is, obscurity is true even within the medical community. At my last annual physical this summer, the doctor was going through my chart and was reading through, you have an allergy alpha-gal syndrome, and I said yes, and we just kind of nodded along. And at the end, I asked her questions about it, and she was like, "Oh yeah, I didn't know what that was. I just was reading your chart." So it's unknown and takes a lot of explanation to really figure out what's going on. But now I'm luckily past that. So that kind of wraps up. I'm happy to take any questions. And I also want to push the Gulf South Vector grant that our director here at the City of New Orleans, Claudia Regal, as well as Janet Hurley at Texas A&M, have this grant for vector education throughout the Gulf South states. If you want to join our listserv, you can scan the QR code there or email cdcvector@nola.gov and we'll get you on. We have a lot of material on different vectors, primarily mosquitoes, but also ticks and fleas and other vectors. So yeah. Happy to take any questions.
Thank you so much to both of you. This is such an interesting topic that I think a lot of people, as you said, don't really know much about. So are there any questions? We'll try to take a couple and see.
I see that in the comments there was 2 questions. One was to ask about contact, although that might have just been to me. But anybody is always welcome to reach out. I'm super interested in this topic and always happy to collaborate. But my email is just john.carlson@ochsner.org. Ochsner is hard to spell, OCHSNER.org. So happy to talk with anybody interested. And then another is a case of someone working in a bee lab who had a reaction to pork after being regularly stung with his occupational exposures with no known tick bites or anything else. You can get alpha-gal by being sensitized through other means. So we're still in the early stages. So this Lone Star stick definitely is our #1 in the US, but it is definitely not the only way. There are actually, it was first identified because of a monoclonal antibody that was mammalian derived that acted as a sensitizer. Since then, Ascaris lumbricoides, the giant roundworm, has been found as a sensitizer in South Africa. So really, really quirky. It's not just these ticks, but it is usually these ticks in the US.
Thank you. We do have one Q&A question. It says, you mentioned you're not experiencing it anymore, but can it reemerge?
Yeah, I think we'll see, right? I mean, I think the reaction time had been over a year before, so I'm still curious to see, as we go on time and time. But yeah, maybe Dr. Carlson can speak more to that too.
Yeah, typically, the only good news about alpha-gal is that it usually goes away on its own within 2 years. And that's why you saw 2 years after his first terrible reaction, Mark came back and tried it again. He wasn't being masochistic. He was actually following the data that the chances were he would have been cured, but he wasn't. And so at that point, you use different statistics and it becomes less likely that you will get rid of it. And so we did a very experimental desensitization protocol called a food ladder. Any food you can theoretically use this for, but there's not really good data on it. And so it worked for him, but because there's not data, we don't know if he stopped eating red meat for a period of a few months, would the allergy return? It's certainly possible, but with ongoing exposure, that's pretty unlikely.
Pretty interesting.
It's tough as a physician. I don't generally recommend people eating bacon every day, just to put that on the record. I don't know that bacon is typically a medical tool for helping people.
Do you want to do one more?
Sure.
While you were in the field, did you take any precautions against tick bites then?
Yeah, that's a good question. And yeah, we did this as a webinar previously, and I had a more of a section on tick prevention at the beginning, but I figured that wasn't as necessary here at the academy. But yeah, in my time there, I was more worried about chiggers because the chiggers were very bad. So I would spray some DEET around my ankles and go out. But I really do wish that I had used permethrin treated clothing or some sort of better tick prevention. I just, always picked up ticks and lived with it. But had I had enough prevention, there was a chance that I would not have picked up alpha-gal syndrome. So I would definitely recommend permethrin treated clothing and other methods. Yeah.
Great. Well, thank you both very much for this. Thank you everyone who had questions. Sorry we didn't quite get to all of them. But we are going to try to stay within a few minutes of our time here. It looks like our next presenter is here. Oh, perfect. Dr. Herrin, how are you doing this morning?
Good. How are you?
Good. Thank you for being here. Yep, perfect. There's our question. And I'll give a little introduction. Dr. Brian Herrin is an associate professor at the Kansas State University College of Veterinary Medicine. He graduated from Oklahoma State University for both his DVM and PhD. His current research focus is on the surveillance of vector-borne emerging parasitic infections and the control of ticks and fleas on companion animals. Some of his recent interests are on early effects of isozolines on ticks and pathogen transmission, surveillance of wildlife for echinococcus. Sorry, I'm hoping maybe he can pronounce some of these for you in a second here. And the surveillance of flea-borne pathogens in his home infestations. Although his research focus is mainly on ectoparasites, Dr. Herrin enjoys working with all parasites of veterinary importance through the diagnostic service at the KSVDL and teaching/outreach opportunities. Thank you, Dr. Herrin. If you are,
Yeah.
are you able to share?
Yep. Should
Perfect.
be able to. Yeah. Thanks for having me on here. I'm really interested to chat about how we think about parasite control for our small animals. Predominantly, I'll hit horses a little bit at the end. And just in keeping with maybe like the integrated portion, talk about what those options or lack of options are when we're chatting about tick control on cats and dogs. And so there's been a fair amount of surveillance on ticks from cats and dogs across the United States. They're super common in presentation for any animals that spend time outside. And I would say also not. We had a couple of cats in the study that the owner claimed were 100% inside, and they submitted a tick to us. So we're thinking, okay, I don't know what that means. And when we're ranking those to see what might be most common, you'll see derma center pop out in both cats and dogs. American dog tick, so that's like helpful that is following its name. Ixodes scapularis, right, the tick that we know transmits Lyme disease. Amblyomma americanum, the Lone Star tick. And then specifically, and terribly misspelled by me there, Rhipicephalus sanguineus, which is a true dog tick, and we'll talk about that one separately. Now, while this paper kind of talked about what was seen, there was obviously massive regional variation. And when I'm talking to veterinarians, a lot of it is kind of who cares? Like why do we bother IDing these ticks? I'll just put them on some, control, whatever. Well, we think about one of the ways to manage this is avoidance, and every year I'll have veterinary students that say, hey, I take my dog on a hike on this nature trail every week. And, starting like 2 weeks ago, we just get 100 ticks every time we go out there. Like, what should we do? And it's like, stop doing that, like just stop. And so there's some seasonality that we want to think about for ticks, and avoidance can be a really effective thing, especially when we're doing like optional things. It's harder when we're chatting with, outside natural resource workers and their job takes them out there, or you're doing a master's and wandering the woods, been there, done that. But if you're doing optional activities, there's times when avoidance might be the correct thing. And so when we're chatting about how do we think about ticks on our pets, our owners perceive them, and they'll see several ticks. It freaks them out and they want action now. And really our focus on tick control for pets is focused solely 100% on the pet that's right there in your clinic. Understanding that when you send your pet out to do whatever activities it is, it's very difficult to manage the environmental ticks in any way. And I always tell people, and this is like, I don't know, back of the bar napkin math, they don't spend a ton of time on a host, and there's lots and lots of stages out on wildlife. And so when we're thinking about treating the 5 ticks on the dog compared to the 500 ticks that are right here on these deer ears, we don't have a significant way to affect the population of ticks focusing just on our pets. That's not so true for other things like fleas. We can kind of mess with the least home infestation or the numbers of fleas in home just by treating the pets. But when we're thinking about ticks, the tick control we're applying to our pets, it literally is only doing stuff for our pets and has no significant impact on the overall burden of ticks out there in the world. And so again, on animal, tick control is the focus of vet med. And again, it's focusly pointed at just dealing with any ticks that specific dog is going to see. We have a variety of options. I think the isoxazolines, kind of mentioned that it's a newer drug class. If you're familiar with things like Credelio, I just have the box there just because. NexGard, Bravecto, Simparica, they're all going to be in the same drug class. They're systemically acting, meaning regardless of if they're taken in orally, there's a topical version, there's likely to be an injectable version soon. It still goes systemic, and we'll talk about what I mean by that. And that's in contrast to, the normal use of pyrethroids or fipronil in our small animals that are topically applied, topically acting. And so there's, very little systemic absorption of those products. The isoxazolines are very effective currently. They're by prescription only, and so it does require you to work with your veterinarian, and that is a barrier to control for some of these pets that maybe don't have a relationship with their veterinarians, versus the topical products just due to them being EPA registered, versus FDA. There's not a prescription required, and you can get them over the counter. When we think about how they're comboed, they're not really comboed to increase or with integrated pest management maybe in mind. The pyrethroids are often comboed with some kind of other insecticide, probably targeting fleas mainly, so that you can use a product for fleas and ticks, right? And that's how most people think about these products, is flea and tick control. And so with the pyrethroids, having pretty low efficacy against fleas anymore, you often see them comboed, even pyrethroids with fipronil. And then you'll add in some insect growth regulators. All that is to deal with fleas. None of that combination is really targeted that much on ticks, other than I would say there's lots of I would say anecdotal, but we'll talk about for specific species. The fipronil can struggle against certain ticks. And so the combo of the pyrethroids and fipronil is just trying to like round it out. That was done late in the game, and it was really more done just to maybe drag along some products that were losing efficacy, and it was easy to just tag in some new chemicals in there. None of it actually had any like integrated pest management in mind. And the companies really struggle with that. They really love when they make an incredible product to just promote the heck out of it and burn it up. And then when they burn it up, they'll combo it with something else and then think about multimodal insecticide or acaricide. None of it is really forward thinking, and that's been like a bane of my research career. Some of the things that it's true for lots of these, but I'd have to chat with my veterinarians and their clients about a bunch, is the topical products, the way they're labeled, they're often labeled as ticks do not have to attach. And so as the ticks move through the hair coat, they're exposed to the chemical, but they do attach. And we know that. And, in the first of the month, they're very quick acting and the concentrations in the fur are high. And so the ticks may want to get off that animal or die very rapidly. The speed can wane over time, though, just as the products degrade due to sunlight or maybe water dilution. And I think one of the best studies showing this, study that Dr. Dryden did, and this is a permethrin product in red and a fipronil product in pink. This is not an efficacy study. This is not how effective the product is. They threw ticks on these dogs. So they threw like 100 ticks on these dogs, on some control dogs and treated dogs. And they counted how many ticks stayed on and tried to attach, and how many were just trying to flee the animal, running away. In a pretty short amount of time. And you'll see the days post-treatment, 3 days, 7, 14, 21, 28. So throughout a month of dosing period, early on in that, the permethrin products do have some, and we'll call it repellency, where 72% of Exodes tried to leave the host compared to controls that were like going down, but you don't see that consistently. So for Lone Star ticks, even at that first time point they used, 3 days, the ticks were not trying to leave the host. They were trying to attach to the host. Those ticks would still be killed over time, right? Again, this is not an efficacy study, but I think lots of people think if I put a topical product on, it makes a magical force field barrier that's just launching ticks away from my pet. And that's just not true. And especially for fipronil, it's not how it works at all. And so we try to talk to our owners about their expectations that they may find ticks attached, and hopefully those ticks are dying in a timely manner, even if it is a topical product. And we know that's true for the systemic because the product is absorbed, and so there's not really any residual on top. Even the topically applied ones, there's no real evidence that there's a repellency on those. And so the ticks do have to attach to be killed, and they have to take in a dosage level of that product to die. And so while it can be more uniform in distribution, there's always concerns that the ticks are feeding, and feeding means pathogen transmission. There's a million products out there, and pet owners are often overwhelmed. And so when I'm talking to pet owners that, either they're not enjoying their tick control, or maybe they are and they just have questions, the best information source is really their veterinarian. And I would promote that 100%, and saying there are products that work in different places. I and different ticks, so your recommendations may be different. My overall question always is, are we killing ticks fast enough to prevent the transmission of pathogens? And I pulled out Amblyomma americanum because I think it's the hardest tick to kill, the Lone Star tick. And you'll see even within a drug class, so this is the first iteration of every company's isoxazoline, and you'll see them labeled. They're looking for a greater than 90% efficacy, and they're trying to get that. So you'll see 72 hours. So the tick in this scenario was on an animal and considered alive probably for 72 hours. That seems like it could be a long time. The way they're set up, they often don't even look at them for 48 hours. So if they're killed within 48 hours, that's probably the minimum of like what they just set out to do. But you'll see there's differences. So this Floralaner 12-week product is great. It lasts for 3 months. But for Lone Star specifically, the label is only for 8 weeks. And so I think that's a tricky spot, because they'll be out there pitching, oh, you only have to remember every 12 weeks, except for if you're in Lone Star areas, you got to remember 8 weeks, because there definitely is some gaps in that. And so we know that tick is hard to kill, and that's different than Exodes. And I often joke that if you want to like convince the public that your product is great, do an Exodes study and say you blocked Lyme disease transmission, and everyone's like, good, hate Lyme, hate Exodes. Those ticks are so wimpy. I could cough on them and I think they'll die. Lone Star, if you want to impress a parasitologist, you're looking to impress me, run a Lone Star tick study, because they're very, very difficult to kill. And so companies have done that. They went out targetedly first to show that you could kill ticks, even though they're having to attach, you could kill them before they're transmitting pathogens like Borrelia for Lyme disease or Anaplasma. Some of these protozoal diseases that take some time to transmit. Cytozoan was a big one. The Lone Star tick's hard to kill, but cytozoan is a very, very fatal disease of cats. And so I think that was an important one to address at some point. But there are studies where these products have failed. And some of our rickettsial agents, like Ehrlichia or Rickettsia, they may transmit quite quickly. And so we don't know if we're blocking that transmission in time. And so in this specific study, they tested out an afoxolaner and a Fluralaner product, and some dogs that were treated did become at least antibody positive to Ehrlichia canis post-exposure. And so now we're really interested in kind of what is that cutoff. I saw a question about the dosing Brevecto every 12 weeks. Is it safe? It's actually labeled that way. So the answer is yes, it's safe, because it has 2 separate labels. 12-week formulation for all of their ticks, and every 8 weeks for Lone Star ticks. So it has been evaluated for safety in that way. So what do we know about like how much isoxazoline does a tick need to take in? Actually, not that much. It's kind of interesting. And we've done some studies here too, but basically if you put a tick on for a short amount of time and then remove that, so the tick was on for 1 to 2 hours feeding on a dog, pull it off and put it back into a humidity chamber to see if it lives or dies, they'll die. And so they've drunk enough of the poison to die eventually. So that's good news. So if a tick is feeding on a treated dog and drops off, it's unlikely to be successful in finding a new host and all those things. So now my interest is really, but what's going on in the meantime? What's happening in that 4 hours? Are they still acting normal, or are they not acting normal? And so I often think about this and think, maybe the normal feeding process is being shut down. you know, maybe, and to be fair to how these studies are run, the FDA only considers live or dead. And so when you check back at 24 hours and a tick barely moves one little leg, the FDA says live tick, right? That tick is different than a tick that's moving around, running, going crazy. And I get that you can't make little distinctions, live or dead. That is fair. But that moribund tick, is it transmitting pathogens in the same way? And so we've been thinking about ways to investigate towards that. And part of that is looking at the musculoskeletal system. So we did some weird things here where we put some ticks on treated animals and then we videoed their ability to move, maybe as a proxy on how they can feed. There are multiple muscles of feeding in ticks. And so we think if their muscles on their legs are shutting down, potentially the muscles of their, feeding apparatus are shutting down. And here, this tick has been on a treated dog for 2 hours and then we removed it. So it only got 2 hours of product and immediately was evaluated. It hasn't been sitting around for very long. And you look at it, it seems to be fairly normal. This is a Lone Star tick. It's roaming around. It's going to start to wander. It's going to get a good carbon dioxide cue because I'm standing down here at the bottom of the screen videoing, and it's going to say, oh, yep, I found a host, and it's going to turn and come straight down to me. I would consider this to be relatively normal behavior for a tick. And again, it's only been exposed for 2 hours and then immediately evaluated. So no waiting around time. Versus ticks that we had on dogs for 8 hours. So this is now a tick that's been on for 8 hours and we removed. And what I like to call this one is like the drunken stagger hone from the bar. It still is moving, but you can tell it's not moving as far. It's not moving as fast. It's not as coordinated, right? And so within 8 hours of being exposed to the product, some of these ticks were already dead and some are in this moribund phase already. And we're hoping that translates to also a disruption of the feeding processes that might be necessary for transmitting these pathogens. So just kind of like a cool look at how we're thinking about assessing these products. Yes, they're effective. Now we want to take it one step further and see, are they effective in a way that really prevents the transmission of these diseases that we're concerned about? And the next big question we get is, well, if you're giving monthly prevention, isn't that a great way to select for resistance? Like, are we concerned about resistance? And I think the issue with the vast majority of the ticks that we talked about, Dermacenter, amblyoma, Ixodes, these are 3 host ticks and they spend a lot of time in the environment. So they have a low host specificity. They're all over the place. So that decreases that likelihood that we're going to be selecting for resistant populations. In terms of compared to fleas that can have a really short life cycle every 3 weeks or so, we consider this a long generation time. They do feed before they mate for most of them. Ixodes sometimes don't, but for the vast majority, they feed before they mate. So that is a selection driver. And they lay a lot of eggs. So that is also a good way to select for resistance. But the way that we're treating these animals, there's multiple acaricides. I will be fair in saying the vast majority of the veterinary market is moving towards the isoxazolines. So that does get to a mono kind of drug use, and that can be pretty heavy selection pressure. But most of our hosts are untreated. We're thinking about all the deer, the rodents, everything, because except for the Lone Star tick, we very rarely see lots of immature stages on dogs and cats. Those are out on the wildlife. And so we're not really targeting multiple life stages with the same drug in the way that lice resistance is a really easy selection because all of them are on host. And when you put a product on, you're exposing every life stage to that product, right? And so this is a way in which there's lots of refugia because there's environmental stages out there. And I think it would be very, very difficult for us to drive resistance with just treating dogs and cats. Also, despite what people think, compliance is pretty low in pet populations, and there's an assessment that probably only 30% of the pet population is receiving really consistent heartworm and flea and tick control. So our practical considerations, we do have people come in and say, the product failed, I saw a tick. And so we need to make sure they understand how the product's working. If it's an isoxazoline, the tick had to attach to get it, and it will die over time. If you see a tick, remove it. But they're freaking out about it. There's also just different formulations. This is a case I saw, it was a Demodex case, actually, so a mite case, but they were unable to clear the mites with Bravecto oral, and these mites are very sensitive to fluorolaner products. And I was like, okay, I don't know what's going on. I think my best bet is maybe try the topical one, and the dog got way better. Was that dog cheek pouching and spitting up the product? Was it vomiting it? Was the owner not giving it correctly? Like, we can't tell. And so from a small animal standpoint, it's really difficult for us to track resistance or decide what's going on. And really the only species we've ever been able to do that for is Rhipicephalus. And there's multiple reasons. It's really the only tick that inhabits buildings. And while it's a 3-host tick, every life stage prefers dogs. So it'll feed, it'll drop off, it'll go hide in the kennel, and it'll come back out as its next life stage. And so you can get these massive, massive populations in a building, all on dogs, all being treated with the same product. Potentially the environment's being treated with the same product. And so it's a really tricky parasite to control. It's something we think completely different than our kind of outside environmental ticks, because you really need to focus on the pet and then find where they are in the environment and treat them, because you get these massive infestations on dogs. There's been documented resistance to multiple drugs in Rhipicephalus, from the permethrin on animal products to off animal environmental treatments. A lot of this comes from, I don't want to say unregulated, but unchecked drug use in things like racing greyhounds, where they had an infestation and they're just spraying the environment all the time with all kinds of stuff. They're not thinking about how do we manage this at all. And so in thinking about the one tick species that I ever think we're going to drive resistance towards, it's always Rhipicephalus, and it's in these big dog kennels or breeding facilities and things that are not taking appropriate care. They're doing things haphazardly. They need a veterinarian or a knowledgeable person to be in there helping them make smart integrated pest management decisions. And they're just out there just spraying the heck out of things. And so it does become a problem. In horses, the situation is even more dire. Like, there's not that much of information about ticks on horses, but when we talk about it and I mention it, every horse owner is like, "Oh my gosh, this is, I hate finding ticks on my pets, or my horses. It happens all the time. Every time I go on a trail ride." And so we set out to kind of mimic that show us your ticks study that I referenced in the beginning. And we went out and looked at ticks on horses. We found the usual culprits again: Lone Star ticks, Ixodes, Dermacenter. We have some ear ticks up here, which are kind of interesting. This giant group down here of Dermacenter albopictus that are very unique ticks. If you've ever heard of the ghost moose tick, or it's a tick that kills moose, there's strains of it that do that. They're kind of a winter tick that we find on horses, and they were a major submission. The data's very, very skewed though, because we're asking for submissions, and one very kind veterinarian took the time to pick 329 ticks off of one horse and send them all in. So that's a huge skew just from one submission. And he commented, "There's more. I just stopped because I got tired." And in fact, this is really a one host tick. And so we see them come up in giant populations in the winter here in Kansas because they're a cooler weather tick. And that's one of the unique things that horses have compared to our small animals or humans. Most people will not experience this, but if you do have a horse owner that's freaking out, "I just came and my horse all of a sudden has like 1,000 ticks on it." Yep, it's this tick. Luckily, even though it's a one host tick, they have a weird seasonality such that one treatment and most owners don't perceive a problem until the next year when they come back out. And so it is kind of satisfying. Again, there was lots of variation in where those ticks are and what the owners are concerned about. And so I know that Ixodes is common in the North Northeast. And when we surveyed those owners, they're most worried about Lyme disease in their horses or anaplasma, versus in the South Southeast. Amblyomma americanum was the big one. And the concern was tick irritation. Just they make these big, gross wounds where the ticks are, and less about the disease transmission. The seasonality in ticks is what you would expect. In horses, it falls the same for dogs and cats. It falls the same for humans. And it's another iteration of why we talk to our pet owners about tick control year round. So we say every pet, every month, all year round. And I talk about that because I'll tell them that I don't find ticks every day of the year, but I do find ticks 12 months out of the year. And so we do recommend that all of our pet owners think about using tick control every month out of the year. What does that look like in horses? Poor, right? It's so challenging because there's not a lot of good data. And even the stuff that's out there that is EPA registered for horses and you can use, there's not a lot of peer-reviewed literature that supports that it's super effective. And a lot of people are using fly spray, right before they head out for a trail ride, and you have to apply that every time you go out. So they don't have the same long-lasting effective products that cats and dogs have. And another thing for veterinarians, now when we have the isoxazolines, veterinarians have off-label use of prescription drugs where we can make recommendations that are safe and science-based, but aren't necessarily labeled on the true labeling. Where EPA, you don't. And that's where we have to be careful with horses and making recommendations like tying cattle ear tags into a collar or something, or their mane. That's actually an off-label use, and owners can do that, right? They can do that, but veterinarians can't make very strong recommendations that they should do that, right? Because that's an off-label recommendation of an EPA registered product, which is illegal, right? And last, we think about dealing with ticks on pasture. It's challenging because you can't really put acaricides out on pasture that easily. There is ways to reduce that shrubbery and cut down the forested area that supports the ticks. But that may be what you want your horses to live in, right? A cool, natural-looking environment. And so overall issue. There was a pasture burning conversation or a presentation. It is an effective thing. It's not something that's done a lot specifically for ticks. In Kansas, we yearly pasture burn for just fire mitigation. And I think overall that can have some effect, but everyone wants it to be something that just removes the ticks forever and ever. And then I think the evidence that shows that's not necessarily true, but it is a way to kind of manage some ticks. Overall, I think about ticks and tick-borne diseases of our companion animals a lot. And if you have specific questions, feel free to reach out to me. I think there's lots of challenges. I see there's some questions about like veterinarians in general making recommendations. I think small animal veterinarians don't think about integrated pest management very much. Our large animal vets often do because they're thinking about fly control management a lot. And if I can loop in some of that tick stuff, then they get it. I think more and more veterinarians are getting a better understanding of the differences in ticks and their pathogens they transmitted. Resources like the Companion Animal Parasite Council make maps that show where dogs are most likely to be exposed to specific tick-borne pathogens. And we've done a good job of tying that to ticks. And so veterinarians are acutely aware that, like, if you're in the Northeast, you're more likely to have dogs exposed to Lyme disease, and that means Ixodes. And so I think the small animal veterinarians are getting very acquainted with the difference in tick species. I think that's a good thing overall.
Thank you, Dr. Herrin. Did a great job of presenting and answering questions kind of at the same time. We have two more we'll try to hit real quick, and then we'll get to our break here. So one is, do you find horses have dermal responses to products?
So Some horses, yes, they can be pretty sensitive. That happens in cats too, more commonly than dogs. I will say the reaction to those topical products is often to the vehicle and not the active ingredient. And so sometimes you'll see, maybe your cat enjoyed, I'm just going to use a brand name, right? Frontline, it's a fipronil product for a long time. And then you found that Walmart has a generic knockoff and it has fipronil and it says it's just like Frontline, and you use it and your cat has a reaction. It's because they've used a slightly different vehicle in that product. And so we see that happen from time to time. And again, usually it's not an active ingredient thing, it's a vehicle issue.
Thank you. And then one last question that says, you did show that some ticks will leave the household pets. Is there any data on how many of those then get on people?
So I think people think that a dog bringing a tick in presents a massive risk to their family. And I think it's pretty low, actually. When you go hiking with your dog and you walk in and you find a tick on yourself, the initial reaction always like, damn, the dog brought some ticks in. It's like, no, you brought ticks in on you because you were out in the same environment. The ticks don't want to leave the host. And again, if there are, topical products that are exposing them and they do make the tick leave, that usually happens early on in the treatment cycle. They're unlikely to go find a new host that successfully. There is some data showing that like they just put ticks in like open containers in various places in a house. And most of our outside ticks, Dermacentor, Amblyoma, Ixodes, can't survive inside. There's just not enough humidity. And so they're not good at living and setting up an infestation in the house versus Rhipicephalus. And again, Rhipicephalus doesn't even like people. So overall, I think the risk of ticks from dogs to people is actually much lower than people perceive. And people should focus more strongly on their own tick control when they're out hiking with their pets.
Perfect. Thank you. It looks like most people have answered our poll question. We are going to take a quick break here and resume presentations right at noon. So Dr. Herrin, thank you very much. Everyone stick around for some great presentations in the afternoon. We'll see you in about 20 minutes.
Yeah, thanks so much.
Thank you. Hi, Roger, are you on?
Yeah.
There you are, perfect.
Can you hear me?
Yes, we had to promote you to a panelist.
How do
Perfect.
I share my talk? Because it doesn't show up a share window at the bottom. Usually Zoom shows share, but it doesn't show that here. So how do I do it?
Give me a second.
Go to more. Click on more, Roger.
Or on more?
Hang
Yeah.
on. You have to, so accept the invitation to be promoted as to a panelist. It says you declined.
Yeah, I declined. Yeah. Listen, it says chat, raise hand, Q&A, or show options at the bottom.
So you have to accept the invitation to be promoted as a panelist.
I
And then
see.
you'll have sharing position, permissions. Do you see the share screen option now?
Yes, I see share screen now.
Perfect.
I have to move this. You see it?
I do not see it.
It shows up on my screen on your system.
Can you stop sharing and try again?
Let's see. Also, it said at the bottom something about subtitles. So it seems to be on your system, but it shows a big number 2 up on, because that's my desktop too.
Sure.
And then the talk is on there, but you don't see it, huh?
Not yet, no.
Well, I don't know how to get to that. Well, let me try this. Now, do you see it?
Nope, not yet. Can you try stop sharing and then share again?
Let's see. I'll have to.
Otherwise, if you email me the presentation or
somehow I have to escape this.
is it possible for Dr. Dooley to share?
Just a minute here. Now I'm stuck on something. I'm gonna have to say
Well,
end show.
I just shared mine.
I can see it that I can see what you were sharing, Dr. Dooley.
Okay.
Does that
Well,
work for you?
well, I mean, Roger, I took most of your slides and left them in my talk. It's just in case you didn't show up. Okay.
Yeah, I changed, but I changed a lot of the time.
Yeah.
So
Okay. Well, so yeah, I mean, I know
wish
you'd
I could
rather get yours in there.
yeah, I wish I could get mine up, but how did you share your screen, Kerry?
I just, I just went to the, to the share at the bottom.
Yeah, that's what I did.
At the bottom and click share. And
And-
then they gave me a bunch of options and then I just picked the one I wanted.
Right. Okay, now I did that just now. Do you don't see it though, huh?
I don't see anything.
Maybe I need to stop sharing. Okay, I'll stop sharing for a second. Okay. All right, I stopped sharing. So at the bottom, there should be a green rectangle, Roger.
Right. It says
An
share.
arrow says
Yeah, it says-
share.
Right, that's what I did. So I
And you
pushed
click that, and then you're gonna get a bunch of another screen. And
Right.
you gotta choose the file that you want to share.
Right. I choose it. Okay, I'm choosing it there. I chose it. And then I'll hit share.
Mhm. There it is.
Now you see it?
Yeah,
Yep.
now we see it.
Okay, all right. It's in the presentation mode now?
Yep, perfect.
Okay,
That looks great.
So
Um,
I'll follow Roger, okay? So, you know.
yeah.
Yeah.
Your mind.
Yeah, so Kerry, I have too many slides, so I may skip some because I've only got 15 minutes until you start, okay?
Okay. Well, you
Yeah.
take what you want. I'll make it work. Okay, go ahead.
Sure. Well, I'll do a little welcome and introduction for you guys. Well, welcome back from the break, everyone. I hope you had a good few minutes. We're jumping right back in with the first presentation of our afternoon. So please answer the poll question. You have a second. And our first presentation is Dr. Kerry Dooley and Dr. Roger Laine from Louisiana State University. Dr. Dooley is the BASF Corporation Professor of Chemical Engineering, Adjunct Chemistry at Louisiana State University, a 1983 PhD at the University of Delaware. Dooley is the author of more than 150 referred publications, patents, and proceedings, and has edited 10 books, worked on projects funded in excess of $28 million, and has consulted for 16 different companies or governmental entities in the areas of chemical catalysis and chemical reactor design. He has served on the executive committee of the North American Catalyst Society and was its meeting chair in 2007. Roger Laine is a professor of biochemistry in the Department of Biological Sciences and in the Department of Chemistry at Louisiana State University, a 1970 PhD from Rice University. Laine is the author of 147 referred publications, 28 book chapters, and 34 patents, and is a fellow of the National Academy of Inventors. Laine has served on more than 30 grant review committees and site visits for the NIH, NSF, and DOE. He was an advisor to the Frontier Research Committee, the Riken Institute, the University of Tokyo for 9 years. Laine has organized 4 international scientific conferences, including chair of a Gordon Conference. As entrepreneur, Laine was co-founder of Glycomed Inc., Anomeric LLC, and Tumorend LLC. His work has included studies on polysaccharides as ABO carriers on the human red cell structures of glycoproteins, cancer therapeutics, antiviral compounds, and recently insect repellents and toxicants. Thank you both for doing this presentation for us. You can go ahead whenever you are ready.
Okay, so talking about nootkatones, which you have experienced if you've ever eaten a grapefruit. It's a new class of safe, natural insect repellents with, I think, a large market potential. Yeah, now it's not changing the slide. Let me see, there you go. So the purpose is to discuss the uses and markets for nootkatones. We're trying to reduce the costs because they're too expensive now to use for insect repellents, because they're used only for flavors and fragrance. We have established a new synthetic method, and we have engaged Professor Dooley in chemical engineering of the new synthetic methods so we can scale up to reasonable cost. It's a major flavorant of grapefruit, used in fruit juice and confection flavoring, so it's ingestible for humans. It's used in the fragrance industry, so it's topically okay. The name of this kind of compounds are called aromophylane sesquiterpene ketones. They are listed. It's listed as a grass compound. It was first isolated from Alaskan yellow cedar in 1962, but it wasn't insect tested at the time. And we and Professor Henderson and Dr. Betty Zhu discovered and patented nootkatone as an insect repellent in 2001 to 2005. So here's what the compound looks like, and I won't just read all the descriptions here. So how we discovered it was a greenhouse owner, Don Heumann, visited Professor Greg Henderson in entomology with a sample of a vetiver grass that he was growing, which seemed to be insect resistant. He asked if we could find out why the vetiver grass was resistant. Well, we just searched the literature and we found that more than 300 compounds had been identified in vetiver oil by Indian scientists. And the major component was vetivone, which was known to have slight insect repellency. Well, because vetivone was not patentable, I asked Dr. Betty Zhu in my laboratory to scan the Fisher Sigma catalog for compounds that chemically resembled vetivone, because there are pictures of the compounds in that catalog. And we tested them on termites because we had a large grant from the USDA to over several years because of the damage in Louisiana from termites. I won't go into this in detail, but there's a huge amount of destruction in Louisiana from Formosan subterranean termites. So I'm just going to show you a simple test that we did for repellency. We made an auger plate so there was some moisture. We put treated sand on one side, for example, Nootkatone treated sand, and then control sand on the other side. We add 10 termites or fire ants. We just put them in a drawer and keep them in the dark 15 minutes. We pull them out, and if they're all on the control sand, it's a repellent at that concentration. If they're all on the other side, it's an attractant, and we had some compounds that were attractants. And if they're evenly distributed, there's no activity at that concentration. Okay, so Zhu found 27 compounds that had partial resemblance of vetivone, and 7 had insect repellency in a termite test. The most potent compound was Nootkatone, which we published 2001 here and patented in 2003 and 2005. And we have a patent which from 2005, which covers arthropods in general, because we tested a lot of arthropods. So we found high repellent activity toward mosquitoes, ticks, fleas. A mouse model there was done by Otsuka and Tokushima, lice, gnats. And the CDC found deer ticks were repelled by Nootkatone in some independent work they were doing, but it was much later. And other insect pests that we found were both repelled, and some of them were killed. We don't know why. Termites, cockroaches, fire ants, and clothes moths, which is tested by Earth Biochemicals in Japan. It's a clear, pale yellow liquid. It's solid at refrigerator temperature. Okay, so here's vetivone at the top right corner. And if you'll notice, and if you smell, the scent of vetivone is kind of musty, woody. And just by changing the location of this double bond here, it becomes the grapefruit flavor, which is Nootkatone. This compound is sold for this amount of money as the flavorant, about 50 million a year. And it's made in a kibbutz in Israel by oxidizing balancine, which is from orange oil, which costs this much. This is the general structure of these aromophalanes. Okay, so in my lab, we reduced the keto group, and it had 0 activity. So we'll say that Nootkatone has an activity of 1, insect repellency, and vetivone about half of that, but it required the ketone group here. We reduced the double bond in the isopropyl group here, and that didn't change the activity at all. We reduced the double bond in the ring, the 110 dihydronootkatone. It was 4 times as active as Nootkatone on termites. And when we reduced both of the double bonds, it was 10 times as active as Nootkatone. So it became a target compound for development and patent. And as I mentioned, 110 dihydro is 4 times more effective, and tetrahydro was 10 times more effective. Low oral toxicity, considered GRAS. Low dermal toxicity. No skin irritation in human testing. 98% Nootkatone produced no skin sensitization in humans. It has antimicrobial properties, that's separate. And the regulatory status is approved by the FDA for food use and was given GRAS status as a flavor ingredient by FEMA. And the Council of Europe included it in a list of substances If you isolate it from citrus, you can isolate it from citrus peelings, basically, mainly from grapefruit and pomelo. That product sells for $8,000 a kilogram. So it was patented as an insect repellent for the termites to start with, and we were the first lab to show and publish that it had insect repellency. Later, I have to say, the CDC erroneously claimed priority. I mean, we already had patents in this area, and so I think we have an interference problem with them. And they involved Evolva, a company that had licensed patents from our use patents from our LSU discoveries. So there's a little bit of a problem with that whole situation. Evolva apparently filed this, and on July 20th, 2020, the registration of Nootkatone for manufacturing use into insecticides and repellents was approved. As a registration really paves the way for manufacturers to develop Nootkatone-based products for consumers to buy. It's a repellent to Brazilian fire ants in several experiments. And so we just use chambers with treatment, sand with Nootkatones in the middle, wet paper on one side, and then we characterize the number of ants that cross at certain concentrations. Here's an example where tetrahydronootcatone was put on 10% on a piece of paper, and we put fire ants in, and they're all dead the next morning. So for some reason, it kills fire ants. For termites, it shows the dose response here. For basically, this is paper consumption, so we just let them eat paper, and then we make treated paper and let them eat it, and you can tell the effectivity. And the comparison of repellency of these three compounds are shown here. Okay, so in a mosquito test, 5% solution of tetrahydronootcatone and isopropanol was applied to the left arm of professors. They won't let us use graduate students for this anymore. And 29% DEET on the right arm. Professors Henderson and Parrish put their arms in a mosquito cage, and the students recorded hits. Immediately, no mosquitoes attacked either arm, but every 1 hour, the professors would put their arms in the cage, and after 6 hours, the tetrahydronootcatone was still active as a repellent. Some mosquitoes landed, but they immediately took off, whereas the DEET arm was getting landing and blood taking. So the effects of 5% tetrahydronootcatone outlasted the DEET. So it can be used in skin lotion, sun lotion, sprays, treated elastic cloth band for your ankle, and a tick would never climb up your ankle. We tested wood ticks. Some ticks transmit Rocky Mountain spotted fever, as we heard earlier today. There's so darn many ticks, I didn't realize how many there were. CDC tested deer ticks. There's a paper on the relative activity of Nootkatone derivatives. Organic synthesis is shown here. The first publication was 2009 on that. And then there were patents, 2006, and we improved the scheme for synthesis in 2019, and so we got a patent in the US and Japan. And this is synthetic chemistry. It's Nootkatone starting material, beta-pinene from turpentine. So you can say it's nature synthetic, because we start with a natural compound. And the first step up here, ozone is used to do an oxidation, and these are the steps that Professor Dooley is going to talk about, and he's working on chemically engineering these to hot. We can make gram amounts. He has to make kilograms, and we use X-ray crystallography to get the structure right. So we've published a number of papers in this area, and we're quite excited about getting it on the market, but the barrier is the cost. Now, there's been competition like yeast mutant biosynthesis, but I'll just go to the bottom here. The results were 208 milligrams per liter. So for 1,000 liters, you get 200 grams. That's commercially unfeasible. The This one was a Chinese effort, and the Nootkatone production was also 3.3 milligrams per liter, 300 milligrams for 1,000 liters. That's not economically feasible. Allylics tried to develop for 10 years yeast production. They sold to Evolva. Evolva tried to do it. They recently laid off 43 workers, and I believe they might have sold their technology to a Canadian company. But they have not produced anything that is a lower cost yet. So I think it's just very difficult to do this in yeast. I think that's enough for me, Kerry. I mean, I've got a lot more slides here, but and maybe some questions could be, are there resistant insects? And the answer is we found one. It was a fly that attacked citrus trees. So there may be a potential for resistance evolution in the future if some insects have already developed it. Okay, that's the end for me.
Okay, Ryan, I'm gonna go ahead and share mine now.
Sounds good. Roger may need to stop sharing then.
Okay.
Perfect. Okay. Looks good.
Do you see mine?
Yep.
Okay. So, I got involved in this because the 8-step synthesis is only going to produce Nootkatone in its current form at about the same price that it's produced now. And so therefore it's going to take a lot of optimization and engineering in order to get the price down for the 8-step synthesis itself. So we started writing proposals back in 2022, originally to the Department of Defense, because the Army said they were interested in, repellents for tick-borne diseases, repellents for ticks. And then finally we got it funded from NSF, and we just got the money in March of 2024. And so we're working on it right now. And if you look at the, if you look at the current 8-step synthesis, where are the biggest changes? Where do the biggest changes have to be made? The 4th step here, which is an anionic oxycope reaction, by my economic analysis, it accounts for about 75% of the cost to make Nootkatone. And that's because it uses potassium hydride, 18 crown 6 ether as a phase transfer agent, and tetrahydrofuran, a lot of it. All of those are incredibly expensive, especially the potassium hydride and the crown 6 ether. So they've got to be replaced. We've got to be able to do that step without using those types of reagents. But there's other steps that can be attacked too. For example, we think that this step here, that steps 2 and 3 may be able to be done without actually separating the products from step 2. We can just go to step 3. For step 1, we think that we can get the temperatures up some. -78 is a fairly extreme temperature. That's the temperature of dry ice acetone. Commercial chillers, relatively cheap commercial chillers, operate down to about -20, -25 degrees. So we're trying to get the temperatures up on this first step. And we're also trying to replace zinc as a catalyst with something cheaper on the first step. This is an alkene oxidation step. And down here, on this step, we have benzene as a solvent. And we got to replace that, obviously. Also, wherever we have dichloromethane, we've got to replace that too, because dichloromethane is being phased out. It's pretty much industrially phased out. It's a solvent now, but it's being phased out even for academia. And benzene's pretty much phased out too. So we've got to replace those solvents with other types of solvents. So that's what we've been working on. If you look at what we've done so far, for step 1, we managed to get it up from -78. We can do it at around -47 right now. And we've managed to replace the zinc reductant. It's a 2-step reaction. First step is with ozone oxidation. Second step, you use a reductant to convert to the ozonide. this ozonide product, and then eventually it gets worked up to the concentrated crude product, nopanol. And you need trimethyl. You can do this without zinc. You can do trimethyl phosphate. That's actually cheaper than zinc, and you can do that. You can do it at a relatively accessible temperature of -15. So we got to get this down. This is -47 degrees right now, but we're going to work on that by modifying the ozone flow and the amount of, the partial pressure of ozone we use to get that temperature up. With the zinc as the catalyst, we're able to get 95% yield on this step. Now for the step 2, we've been doing work on that too, and we've been able to substitute sodium hydroxide instead of potassium hydroxide. That's going to save some money. And we've been able to take nopanone that's not separated or purified and go directly to step 2 and do the aldol condensation in step 2. So that's going to save some money too. Right now we're getting, you want you want to get the E isomer here. We're getting about less than 20% of the Z isomer. We can separate out the Z isomer and possibly recycle it, but that's about the same yield that we're getting in the patent. Okay. Now, what about the 4th step, the key step? That's an anionic oxycope reaction, and we wanted to be able to do a large number of test reactions using different types of catalysts for this. So what we did was, instead of starting off with the actual reactant we're going to have here, we said, let's try getting a reactant that's similar to what we would have as 8-step synthesis, but have similar chemical characteristics, but is something that's a little more accessible. We can make a lot more of it, so we can run more tests with different catalysts in different conditions. So we came up with this molecule here, which I'm just going to give you an abbreviation for it, called PPEH. Okay, and this has a lot of the same characteristics as the molecule we're actually doing the AOC reaction on in the 8-step synthesis. So we're converting this PPEH to this enone as a product, and we can make this PPH very easily. We can start from dibenzyl acetone and just do a Grignard reaction to make the PPEH. Dibenzyl acetone is commonly found in sunscreens. So we made a large amount of this material, and we're running a lot of test reactions using different types of catalysts, solid heterogeneous catalysts, as opposed to potassium hydride, 18 crown 6 ether, and TA. And we're trying to get away from THF too. So some of the work we've done on that so far, there's the PPH reaction to the enone. And we said, let's start off with some much weaker base catalysts than potassium hydride. Potassium hydride has a pKa of around 34, 33, 34, somewhere in that region. But, I did some theoretical work, and I don't think you need that high of a pKa to do this reaction, and that agrees with some of the recent literature on the AOC reaction. So we start off with some weaker base solid catalysts: potassium fluoride on alumina, cesium carbonate on silica. We've been testing them in different solvents, and we found out that we could get away from DCM and THF. We can do these types of reactions. We can use tert-butyl alcohol, which is a much more benign solvent for this type of work. And we've been able to get reasonable yields. These are actually pretty comparable to the yields in the patent for the AOC reaction in the patent. One of the problems is that when we look at the product that we're getting, the enone product, it matches the proton NMR data from the literature for this product, but it doesn't exactly match the carbon-13 NMR data. So we're having to look into that a little more carefully. Anyway, we've done work with these kinds of catalysts. If it turns out we need stronger base catalysts, we can go to medium basicity catalysts like diazobicyclodecanone, DBU, and support that on cereal alumina, or 2-cyanopyridine on cereal alumina. they give pKa's a good bit higher, several orders of magnitude higher than these catalysts do. And if we really need to go to something similar to potassium hydride, but as a solid catalyst that can be more easily recycled, we're making some polyphosphazines right now. We're synthesizing polyphosphazine, and we're gonna support the polyphosphazine in a hierarchical silica so it doesn't get leached out. And we're going to test that as a catalyst too. So anyway, that's about as far as we've gotten. Of course, we got to scale this up to a little bit bigger size. I mean, right now we're doing everything in hoods, and the only way to do that is to eventually put it on a skid and do it in more things like 5-liter glass setups. So we've got a skid available, and we're buying the glassware and trying to put together the pilot plant on the skid. And the goal is to produce 120-gram batch of Nucatone on the skid, starting from 100 grams of beta-pinene in every 2 days. Okay. So just to remind you, the ultimate goals here are, if we can get this thing down to $1,500 per kilogram, we can capture the flavor and fragrance market. But our real goal is to get it down to less than $500 per kilogram and make it competitive with permethrins and DEET as insect repellents. And once we do that, hopefully we can use the Nucatone product in any of these types of delivery agents in order to make it a commercial product. So in summary, the Nucatones are safe, they're potent insect repellents, they have a wide variety of resistance to old and new mosquito and tick spread diseases. The current market leader for this insect repellency is DEET, but DEET has some side effects that we didn't talk about, and it's used at very high concentrations. We think we could use Nucatone at much lower concentrations and get equivalent repellency. And there's plenty of opportunities for optimization and cost cutting in the 8-step synthesis, and we're working on those opportunities right now. So that's it. And if there's any questions, I'd be glad to answer them also.
Thank you, Dr. Laine and Dr. Dooley. It looks like we do have at least one question. If everyone could please answer the poll question while we start looking at questions here. But the question in the Q&A is Nucatone considered organic?
Yeah, so we have tested most of this as in applications to to, like, for example, in the fleas and the mouse study, we just use isopropanol with 5% Nucatone. It's the simplest kind of lotion you can use, rubbing alcohol, right? And so we tested mostly in that, but it could be put into sprays or skin. You could add it to sunscreens or skin lotion. I have an idea that a cuff around your let's say an elastic cloth cuff that you put around your ankle that's got Nucatone 5% or 10% on it, and no ticks are going to climb up your leg. I mean, you could also treat your socks or something like that, but there could be something sold that just a little elastic band that would be a preventative. But mainly you could apply it as a skin lotion.
Thank you. Is there any other questions? If not, we can transition to our next presentation. Dr. Laine and Dr. Dooley, thank you both very much.
Thank you.
Yeah, thank you.
And our next presentation will actually be a panel discussion. Those of you that were here yesterday will be slightly familiar with the concept. It'll be pretty similar to what we did yesterday. So just give us a second to get situated and we'll do a little bit of an introduction. It looks like all three of our panelists are here. You guys can actually turn your cameras on if you would like, just so people can see your faces while you're answering questions. But if you're here yesterday, you're familiar with how this is going to go. If you're new, welcome. But basically what this is going to be is we have a panel discussion on mosquito abatement districts as a potential resource for tick control. We have three experts who all work in mosquito control, and we have a couple questions for them. But they're also here to answer any questions you may have. So over the next 20 minutes or so, any questions, I'll ask them all to do a brief introduction of who they are and what they do. And feel free to ask them any questions about mosquito control resources and tick control resources. So our panelists are Scott Larson, who is an entomologist at the Metropolitan Mosquito Control District. He's also a co-PI on the tick IPM working group. We have Mark Clifton, who is the executive director of North Shore Mosquito Abatement District, and James Burtis, who is a biologist for the Center of Disease Control and Prevention in the Division of Vector-Borne Diseases. Looks like we're having a couple technical issues. Just bear with us for just a second. But if one of you is able to start with an introduction, that would be great.
Sure, I can go. This is Mark Clifton. And so I'm currently the executive director of the North Shore Mosquito Abatement District. And for those of you who aren't familiar with our organization, we're located in Northfield, Illinois, just north of the city of Chicago. We cover about 70 square miles of kind of suburban Cook County. We have about 330,000 residents, and we have the cities of Skokie and Evanston within our district. Our main function is mosquito control, public health mosquito control. And in the last maybe 5 years or so, we've become much more involved in tick surveillance and tick monitoring as the abundance and richness of species of ticks has continued to increase in Illinois. So I think maybe I'll pause there. Unless you want more of an introduction, I'd be happy to go on.
No, that's a great place to start. Scott or James, either of you wants to.
Sure. Yeah, I'm Scott Larson. I'm the assistant entomologist at the Metropolitan Mosquito Control District. We're a large district centered around the Twin Cities of Minneapolis and St. Paul. Our service area is about 3,000 square miles. We serve about 3 million residents. And yeah, we're mainly a mosquito control agency, but we also work on black fly control. And we have been actually legislatively mandated to consult and cooperate with the Minnesota Department of Health in developing management techniques to control disease vectoring ticks. And that was in 1989. But of course, there is no current wide-scale economically or environmentally suitable method for tick control at the moment. So we're focused just on surveillance currently.
Thank you. James?
Yeah, hi, my name is James Burtis. I'm a research biologist at the CDC in the Division of Vector-Borne Diseases. Our team focuses mostly on ticks and tick-borne diseases. My research has been focused on kind of developing new interventions for tick control, but also kind of the social aspect of tick control, the logistical side, which historically we've kind of left to the wayside. Tick control is a really complicated socioeconomic framework to work with. So trying to figure out who's going to actually deploy tick control in the field at large scale, what the public is willing to allow people to use as we start to kind of develop these new methods that hopefully will be coming down the pipeline in the next decade or two.
Thank you. As I mentioned before, for everyone here, please feel free to put any questions you have for our panelists in the Q&A. We'll start with the first one from Bob, which says, are you employing natural or botanical chemistry in your mosquito abatement programs? If so, what are you finding to be most effective in terms of efficacy and residual?
Want me to go?
Go ahead, yeah.
Okay. So I guess in terms of mosquito abatement, most of what we use would be microbial products. So Bacillus thuringiensis, Israeliensis, BTI, Bacillus sphaericus, and I probably also spinosad, which would be I guess derived from a soil bacteria. It's another kind of naturally derived product. So they're all really effective for us, if long as you don't develop a bunch of resistance, which we've found to Bacillus sphaericus here. But some of them will last, depending on how they're formulated, you know, and what the use pattern is, you know, 8 weeks or more maybe. So yeah, I think it's pretty common, at least in the Midwest region, to employ, you know, natural or microbial products. As far as botanicals go, I don't think there are any botanical products. There isn't much available for mosquito control. I think there are some adulticides that are pyrethrum based, so they're extracted from actual plants. That would maybe be considered a botanical, but a lot of the botanicals end up as 25B products with EPA, and so I just think abatement programs, or at least us, tend not to use 25B products because they don't have the efficacy data necessarily to back them up. So we tend to go with actually FIFRA registered products, not exempt products. So I'll stop there and let someone else chime in.
Yeah, basically exactly what Mark said. We're using the same products in the same way as far as natural and botanical chemistry is concerned.
And just to add to that a little bit, since 25B active ingredients came up, there have been some fairly recent evaluations of some of the 25B active products as both repellents and acaricides, and their efficacy is pretty variable. So, you know, that information is kind of being generated currently. It seems that they're not particularly effective. They have short time frames for the most part when they are effective. So just to add kind of the background there.
Thank you.
Any viruses that infect mosquitoes or ticks? Well, there's a bunch of viruses that infect mosquitoes or ticks, and some that infect both, I believe. So for us, I'll speak to our biggest public health concern, and that would be West Nile virus and Culex pipiens mosquitoes. That's our #1 issue here in the Cook County, Chicago region. We normally would have dozens of cases of neuroinvasive West Nile each year. I think we're well into the double digits for Illinois so far, and they keep trickling in as the health professionals and CDC kind of document the cases. The district here will see, you know, 5 to 10 most years. We've been really effective the last 4 or 5 years, and this year so far we have no neuroinvasive cases, but it's taken a lot of work to get to that place. Ticks, you know, oh, are there any? Oh, I'm sorry, Roger. For control, none that I know of. That's a much simpler, more boring answer.
That's okay.
Just to add, we do have a fungal product that had some promise. It was Met 52 was taken off the market, and I think it might be coming back as I think a Canadian company may have bought that product I heard recently. But that's the only biological that I know of that showed any efficacy in the field. And even then, it's expensive, and efficacy is not as long as you see with the synthetic products. So yeah, kind of the same story.
We have a good question here. It says, do you have any methods or tips for conducting tick-related outreach with the public? We always have a disconnect on what a tick is and what to do if someone finds one. So drawing on your experience with mosquito outreach and awareness.
Sure, I guess I could start. We do incorporate, well, since there's really no way of controlling ticks for us, we adhere to an IPM methodology for mosquito control. And at the base of that IPM pyramid is outreach and education and different cultural practices people can take to avoid mosquitoes or ticks. So that's about as far as we can get in the tick management realm, is outreach and education. So it is a part of our outreach. Our communications manager does talk about ticks. We've constructed like a tick safety kit, we call it, that has our logo on it, and also has some resources inside for tick removal, like tick keys or tweezers or different tools that are available that we can get printed with our information. Also, like a little tick ID card that the Illinois Department of Public Health publishes. Different repellents and maybe a magnifying glass or some other tools, and so we hand those out as an outreach tool, and it also kind of opens the door to conversations about ticks and how to avoid ticks and how to use repellents and all these things. And they've been extremely popular, the mosquito and tick safety kit. So that's one way that we've been able to have those conversations with people.
Yeah, and up here in our district, we also offer like tick ID services. People can bring physical ticks into the lab and we'll ID them for them. They can mail them to us. They can take a photo and try to send it to us that way. We do have a Facebook group and Instagram. We're always promoting, tick awareness. We do put out signage at dog parks and natural areas, warning people about the presence of ticks and the potential for tick-borne disease. We also, we're involved in like all the county fairs and the state fair. We always have booths at those with, a lot of outreach materials. We participate in parades and radio shows. We also have materials like tick ID cards. And then we also work with the local parks departments, state parks, Fish and Wildlife Service to survey ticks on their lands, send those ticks that we collect to the CDC and test those for pathogens. So we can update individual parks on the presence, absence, and abundance of certain tick species that are there, and certain pathogens that are prevalent or are there in the certain parks. We can just let people know that those have been discovered in certain locations. But yeah, it's a lot of outreach, and I haven't found a big disconnect with people and knowing what a tick is. I mean, we're in Minnesota, ticks are really pretty common. Most of the public is familiar with them.
Yeah, so a question is if we have any data, and I gotta say, I think one of the biggest shortcomings in mosquito and vector control is a scientific approach when it comes to public health communication on both mosquitoes and ticks. There just isn't much data on any of the messaging that we use or how effective it is, and that's true for tick kits. We don't have any data. It's a whole, as you can imagine, a whole other complicated thing to assess people's attitudes and feelings on some of these, and I think we're just way behind. So much what we do is science-based when it comes to controlling mosquitoes, or but when it comes to the public outreach, I think there's a glaring gap there in our scientific approach, to be completely honest. As far as the question on sterile males or Wolbachia, there is no Wolbachia methodology for Culex pipiens. And I know people who have explored sterile male methodologies, but and as far as I know, aren't any that have been validated or tried in a big way. It's something we'd be interested in, the sterile male thing, but it just has never happened for Culex pipiens.
Scott or James, do you have any insight on the last two questions? So data collection related to tick kits and the release of sterile males, etc.
So nothing on sterile males. I'm a little out of my field there, but yeah, it is a challenge. So this is something, there have been a couple of recent surveys of publicly funded vector control operations in the upper Midwest, Northeast, and Pacific Coast. That, they suggest that, and it makes sense, that educational efforts are kind of the primary intervention that these agencies can offer. But evaluating that is really difficult because it's very diffuse. Once you get that into the public, it's really hard to tell if that actually has an effect on behavior, or if you want to get even more diffuse, than if it actually has an effect on human incidence of disease. That gets very hard to track. So there isn't great information about the efficacy of these efforts, but they are pretty widespread. We saw a lot of agencies offering, and I think both Mark and Scott likely responded to the survey, offering these services currently. It's something that we need to consider evaluating. It's just how to do that is complex.
Thank you, James. Scott, anything you'd like to add?
Nope.
Sounds good. Another question here, it says, have any of your districts found any novel species or trends in populations recently?
You want to go, Scott?
Yeah, I mean, we typically just have the black-legged tick and the American dog tick here are our main human biting ticks that we deal with. This last year, we did have a larger number of Lone Star ticks collected and submitted to us than normal. Maybe about 30 adult Lone Star ticks. Typically we maybe see like 5 to 10 a year. I'm pretty sure we're too far north for them to be established anytime soon. But there is, the occasional influx of the Lone Star tick. Otherwise, yeah, we get some of the less common Exodes ticks. We do animal trapping as well as tick dragging, but nothing particularly novel or, no big changes in trends in populations of ticks for us.
For us in the Chicago area, there has been some changes. Ticks in general used to be kind of rare in Illinois. And it's just, I think, a lot of different species, the abundance has exploded. Dog ticks are an incredible nuisance for us, and they continue to increase. We've seen deer tick populations increasing. We've seen in the state, Lone Star and Gulf Coast ticks continuing to increase, and we expect them in Cook County. So that's part of why we do these tick surveillance, is to try and create a baseline and look for the introduction or the establishment of populations in our area. And then also the Asian longhorn tick has been found in Illinois, so it'll probably spread throughout the state. In terms of mosquitoes, Aedes albopictus in the last 3 or 4 years has absolutely exploded across our district. We're probably at the northern end of its range. It used to be a mosquito that we would occasionally see, but we see it reliably now. It's probably the third most abundant mosquito, has displaced another invasive Aedes japonicus. So I think in both ticks and mosquitoes, we've seen an increasing number of species and their abundance. But I'm sure James has got even more insight on this question.
I'm gonna again parrot what you guys are saying here. We're seeing this in real time in the surveillance data we're collecting. We're seeing both in the human cases and in our direct tick surveillance data. More and more counties are kind of lighting up with Exodes scapularis. Our team mostly tracks Scapularis and Pacificus. Pacificus isn't expanding so much, but Scapularis, we've seen it expand through the Northeast and upper Midwest. Some of that is probably enhanced surveillance. People are looking more now, but a lot of that is likely a range expansion of the tick. So we have a lot more people at risk. Just incidentally, I got to see it in real time to some extent. Living in central New York, moved there 15 years ago. You couldn't find a tick. I tried, and now it's pretty heavily infested. So, we are seeing these these geographic expansions. And then you also have the Lone Star tick, being detected much farther north than it has been previously. And we don't have as much surveillance on that species, but, we're seeing it in Southern Connecticut. I've heard reports of it in Michigan. You're seeing it in Illinois, Minnesota. These are states where historically we haven't really seen that activity, and that's an aggressive species. So people get very concerned about it very quickly.
Thank you. We have another question that says, are any of your programs currently performing any control measures for ticks? And I'll add to that, are there any existing mosquito control methods that you think could be useful for ticks in the future?
So this is an area we've been active in. There are no real control measures for ticks, maybe a barrier treatment, but that's really focal, and that can be used for mosquitoes or ticks. I don't know that they've been really linked to positive epidemiological outcomes and different studies that have looked. So we don't tend to employ barriers. I don't think we could really employ them in a public wide area format in a way that would be effective. It's just a yard or a something here or there. So I think really there aren't any control measures available. I don't think there's anything that we currently do other than maybe barriers that could have any measurable impact. ULV, the way that adult mosquito control is designed to work, is not likely to impact ticks at all. It's meant to impact flying mosquitoes at night, not terrestrial ticks crawling around. So I don't think that's going to work. We've been exploring adopting kind of a mist blower and adjusting it in a way where we can get a mist that can go out maybe 100 feet and come down onto the ground, kind of like wide area larviciding. We don't have an active we could use in that way, but we would like to develop a method, and then maybe an active will come along that would be sufficient for this. So it's a chicken or the egg. You have to have the material to use, and you have to have the methodology. But if you don't have the methodology, why would anyone invent the material that could be used that way? So we're hoping by inventing a methodology, maybe there'll be we can make a method where you have 100-foot band, maybe at campgrounds or parks or something, that would exclude, repel, or maybe kill just ticks. But it's still really much in its infancy. So that's as much as I can link mosquito control to any sort of tick control.
Yeah, I completely agree with what Mark's saying. I do think we're trying to set ourselves up with our surveillance efforts in an event that some product does come along that offers the opportunity to do tick control, where at least we'll have some information on previous seasons and locations on tick abundance. And yeah, so maybe in the future we'll be ready for it, but yeah, right now there's really nothing.
Yeah, and that's mirrored on the national scale. You have maybe 10% of districts appear to be doing some kind of tick control, and it's usually very limited in habitat management or some barrier sprays with acaricides. Not a lot of large area wide control, because there is no method for large area wide control. It's part of the problem we have right now, is we really have somewhat reliable methods for spot control. You can maybe control ticks moving into a high use public area, but you're not going to get at that base of that population the way you do with mosquitoes, where you can really knock it back right now with the methods we have. And there's also the cost effective side of it, where even if we do, the methods we do have that might target deer are going to be very expensive and require a lot of budgets to increase pretty substantially to actually be able to use. And then there are regulatory issues with, and I see that that's actually coming up as the next question, with the use of synthetic acaricides. We have a trial ongoing in New Jersey right now, trying to look at the cost of applying multiple interventions on public land, and it ends up you almost can't apply the acaricide anywhere because there are regulations governing distance from wetlands, distance from residences. So there are a lot of roadblocks even to our simplest interventions. I'll let you guys take over.
That's completely accurate. It's just a fundamentally different problem than mosquitoes. At least with mosquitoes, you know, if you can identify that larval habitat, you know where they're coming from. They have that weak point in their life cycle. But ticks, that's not that's not possible, really. And so I don't, you know, in terms of our communities being willing to allow wide area control of ticks, I do not think so. I think that would not be a terribly popular. I don't think people would see at this point the cost benefit, you know, where that would be something they'd be willing to accept, because we just don't have a technology to do it in a way that makes sense. Maybe if there's some other technology, or, you know, that was out there, perhaps that was something tick specific, you know, that didn't have any non-target. But, you know, right now, I mean, everything available isn't specific enough for ticks to make that work.
Yeah, and I'd say, oh, go ahead, James. Okay. Yeah, I'd just say, you know, for us, I think maybe a biological product like Met52 might be acceptable to the public, but even then, I know we would have pushback. You know, Minnesotans in general is pretty environmental and not, you know, we don't do very much adulticiding for mosquitoes even up here. We get pushback from whole communities. Minneapolis Parks Department, for example, you know, is banning basically all pesticides, pesticide use in their parks. Yeah, so I think there's a lot of hurdles to applying that, and yeah, I would wager a synthetic acaricide would not be accepted by our residents.
Sure. I'm going to piggyback off that. And yesterday we heard from a grad student who's doing some research on the use of prescribed burning. I just am curious about what your opinions are on that. If you think that's like a good control option, and something you think could be accepted by kind of the public.
I don't know enough about it in terms of ticks. I know that our Forest Preserve District does prescribe burning, but for a lot of other management reasons. I don't think it's specifically for ticks, and I don't know that anyone or they have even studied how that works for ticks.
Yeah, there is some research on prescribed burns. They do seem to be somewhat effective. I think we probably need more research I think those were mostly in kind of more isolated forest habitats. So we'd have to see kind of if it would work in a more, you know, populated area. As far as I know, you know, there isn't a ton of research. There is some evidence of efficacy. I think it's a matter of figuring out how you do that in a place where people are actually at risk and not in a more natural area. And I just want to also mention this is something that's come out both in our survey of vector control operators, but also we put out one recently looking at public lands, so tick control, what's happening actively on public lands, what interventions they might be interested in. And these kind of more mechanical interventions are pretty popular as an option, I think, because there are fewer regulations around their use. So, you know, things like controlled burns, path widening, vegetation removal seem to be some of our more I guess I would say more politically viable interventions that we have right now. So I appreciate that question.
Yeah, I would say that, you know, most of the prescribed burns up here are also part of ecological restoration or removing invasive species, that kind of thing. And so, you know, you might get some overlap of some tick control in those areas, but, you know, in general, I don't think we would be doing very large area burns in this metro area or in the, you know, adjacent wetlands and forests that we have. So yeah, I could see it too somewhere, but probably not on this scale.
Oh, sorry.
Well, go ahead if you have just one more thing.
No, I just saw the question about host. Well, it's gone now. About host targeted interventions and wanted to jump on that. But did you have a?
we're just unfortunately out of time for
Oh.
additional questions, but this was really great. I think we had some really good questions and some really interesting answers. So I want to thank the three of you for being willing to share your knowledge and being here today. And I hope you stick around for our one last presentation here. So thank you very much.
That was fun. Thank you.
Yep, thank you.
Thank you very much.
Thank you. We will. Oh, looks like we're all perfect. Okay. So our final presentation of the day and of the TIC Academy here is upon us, and that will be Joellen Lampman, who is the Community IPM Extension Support Specialist with the New York State IPM program at Cornell University. With a degree in natural resources from Cornell University, Joellen is a lifelong environmental educator and learner. She is currently pursuing a master's in science communication at Stony Brook University at the New York State IPM program. She utilizes the clear knowledge-based decision-making process of IPM to teach ecology and make a difference, one property at a time. In some circles, Joellen is also known as the Tick Lady. So Joellen, thank you so very much for being here. If everyone could take a second and finish answering the poll question. And Joellen, the floor is yours. Thank you.
Great. Well, thank you so much for inviting me to talk about our tick education program, the Don't Get Ticked in New York campaign, and for hanging in there to the very end of the TIC Academy. I'm really passionate about this project, and I am grateful. I just want to put it out there. I belong to this amazing team that we worked really close together to make it happen and to continue to improve it over the years. So here in New York, we're endemic to not only Lyme disease, but other tick-borne diseases, especially those spread by the black-legged tick. These maps from the CDC show the distribution of these pathogens throughout the state, but the different regions are not evenly impacted. As James mentioned earlier, in central New York, not too long ago, it would have been hard-pressed to find a black-legged tick. And it's a lot easier now throughout the entire state, but there are still regions of New York that are getting used to that. And people that are having a hard time in Western New York, convincing their healthcare providers that they should get a test for Lyme disease. So we had been providing tick education for many years, but in 2017, the New York State Senate Task Force for Lyme and Tick-Borne Diseases provided us with funding to create a specific tick education campaign. And we used those funds to create the Don't Get Ticked New York campaign that launched in 2018. So in preparation for creating this campaign, we looked to see what was already out there. And what we found was a lot of misconceptions, a lot of misinformation. We also found lots of questions, and we did find a lot of text-heavy information as well. So we set out to take on these challenges in the development of our new campaign, which is just a scattershot of a lot of different methods. The primary goal is to reduce human exposure of ticks and tick-borne diseases to make tick avoidance easy to understand and accomplish. We use those funds mostly to hire a graphic designer to help with the visual aspects of the campaign and to invest in resources to better communicate science-based information. So we had to make some decisions on what to cover and what not to cover. So we decided that identification was a really important piece that we wanted to touch on. It's probably not surprising to you, but we have found that many of the people that we talk to don't recognize that there is more than one type of tick. For example, a lot of people think that black-legged ticks are just baby ticks. And this identification is really important because, as you know, these different ticks do have different health risks. So to that end, we created a bifold identification card with the 3 primary ticks of human health risks in New York. We included their sizes because we feel that's really important. the instructions for doing a daily tick check, tick removal, and the next steps to do after they've been bitten. And we have these cards in Spanish. Now, right after we had these designed and we printed out a bunch of them, here comes the longhorn tick. I just want to point out that we've made the decision in anticipation of the ESA changing the name. We are now referring to the Asian longhorn tick as the longhorn tick and taking out that Asian part. So this guy moves in, or I guess gal moves in, and then very recently the Gulf Coast tick has also climate migrated into New York and is established in parts of it. So it's time to do a new tick identification card, and we released that this year, and we decided we needed to go to a trifold. We didn't want to reduce the size of the images of the ticks. We wanted to add these new ones. And this also gave us an opportunity on one panel to put on a QR code so that people can easily get to our website, both to the page that talks about how to protect yourself from tick bites, as well as a web page that we have dedicated to the health risks of these different ticks, which is the reason that we're talking about identification in the first place. So within that page, we show these 5 species as well as the potential health risks for them. And this is regularly updated. The last time it was updated was in April of 2024. I hope it won't need updating soon, but I suspect that it absolutely will. We have done training for master gardeners across the state that have people come in with different specimens for identification. So we've taught them how to do tick ID. We also provide them with this resource with the idea that once they ID the tick, they're going to circle the species and the life stage that came in. And then the client can take this to their healthcare provider if they have questions about the tick bite that they received, because our job ends with what to tell your doctor. And we've received some really great feedback that the doctors are really appreciating this resource because it's providing them with information that they don't know. And I have the QR code if you want to go directly to that web page. Keep your phones out. Got lots of QR codes in this presentation. So in addition to focusing on the identification, we felt it was very important to also address the size of ticks. Educators often tell people to conduct a tick check, but people don't know how small they are. So we created tick education kits that included ticks in vials, and those vials are filled with hand sanitizers, which allows the ticks to be in there floating and not sloshing around, making them easy to see. We also borrowed from the wonderful Tick Encounter education program, their idea of tick bagels. So you want strange looks, you go and you ask for 119 poppy seed bagels, which I then dried over and over again and then glued the ticks onto them. If you drop one, it'll shatter like glass, but they're still lasting all of these years later. And people really, it's a great way for them to really visualize and get that search ID in their brain of what size they're looking for. We also had tick tattoos printed out in both the adult and the nymph size, which is really helpful for getting that point across when talking to people. Also, when we do education with children, we like to send them home with the tattoos to help reinforce the lesson with their caregivers when they get home. And we were also able to get these field cards from Brigham and Women's Hospital. They have a slight indentation on them so that you can feel what a tick might feel like if it was attached. Because they're so small, because there are parts of your body you can't see, we really encourage people we're talking to not only look for ticks with your eyes, but also to look for them with your fingertips. So we created these tick education kits. We distributed them to Cornell Cooperative Extension offices across the state. Educators also received a supply of identification cards and tattoos, as well as instructions for how to use the kit and links to different fact sheets. We also created a PowerPoint presentation that's available to extension educators on a shared file. We can't do it all. We're just a few people. New York is not the biggest state, but it's pretty large. But we can do our best to make sure that people that are also out there doing TIC education have access to our vetted materials. And of course, we're focusing on personal protection. And one of the strategies that we used was to create a series of infographics with the idea to communicate one or two key ideas and make them really visually accessible to people, for people that English might be their second language or they have low literacy. We really wanted these to be less work to understand the concept. We believe that having materials that are targeted for specific audiences leads to greater attention. So we created a series of these daily tick check infographics targeting farmers, hunters, children, gardeners, anglers, campers, farm workers, golfers, hikers, and soccer players. And if we have more audiences that need it, we get more funding, then we'll look at expanding this list as well. We created infographics for the personal protection strategies, permethrin-treated clothing, repellent use, which includes on there those that have active ingredients that are registered by the EPA and have been shown to be effective against ticks themselves, and also one on protecting your pets. And we also have some that are to address tick management on school grounds and backyards with a real focus on the monitoring and how to do that. For efficiency, we take these images off of these infographics and we use them in multiple ways in our PowerPoint presentations, or this is an example of our pull-up banner that we use when we go out for tabling. And you'll notice that we include multiple strategies for protecting yourself from tick bites. And this is something I've been playing with. So one of the things that came up in the pandemic was this Swiss cheese model for multiple ways to reduce your chances of getting COVID-19. I thought this was a perfect example for using for tick protection as well. So this is an image that I use in PowerPoint presentations a lot. And then I also made a hard copy, if you will, and you can see my coworker Regina there holding it. This allows us to talk about the different strategies and why they work and why they might fail, and then very visually show that they're going to fail in different ways. So if we layer them up, we're going to close up those gaps and reduce the risk that you're going to get these tick-borne diseases. Really driving home the point that there's no one thing that you can do to reduce your risk. You need to do multiple things. My latest iteration is a riff on cornhole. So I have this tabletop cornhole game that I made the hole much bigger, and using those same boards, if we lay them across, it makes it harder and harder for people to score or the tick to get to the blood meal. Still working on this. If you have any other ideas, contact me. Let's collaborate. I think I really got something here. Speaking of the pandemic, when we weren't able to get out and do in-person events, my wonderful colleague, Matt Fry, created 4 videos featuring Dr. Fry, entomologist and tick educator, where he is working directly with Maddie. And Maddie just needs tick education to protect himself from that tick bite. He created 4 videos on dressing the parts of the long pants, tucking them in, you know the drill, how to check for ticks, the use of repellents, and permethrin-treated clothing. They're about 2 minutes each. The first one has over 3,000 views, and I might be biased, but I think they're pretty wonderful. Also, in the video realm, we created let me pause this for now. We used the funding from the original grant to create, to contract with then graduate student Max Helmberger to do this claymation video for us. And I do want to just play a little bit of this for you. This is what So you can see that we do have real science-based information within that infographic, but it's kind of in a fun way. It's received almost 13,000 views, and the comments are filled with praises for how this is very kid-friendly, a great way to get this information out. And we find that unlike us, we admit we're kind of a strange bunch. We don't want to, most people don't want to see live videos of actual ticks like digging into skin. So the claymation just makes it friendlier and more palatable for a lot of people. Another pandemic thing that started was the What's Bugging You webinars that was put by Dr. Matt Fry and Dr. Amara Dunn-Silver. These are half-hour webinars that are the first Friday of every month. They last for half an hour. It consists of a 20-minute presentation on a larger topic and then a 5-minute quick glance at a different topic. Next month, November 1st, we're actually going to be having somebody from the Northeast Vector-Borne Disease Center of Excellence come to talk about the Tick Blitz project, a community-based project looking to assess ticks across the state. I recommend that you join in if you want to learn more about that project. And we help to collaborate with that. And of course, we also have our website, which has, we were so happy to get this handle, don'tgettickedny.org. Beyond our static stuff, we also, of course, are out there trying to reach people as much as we can. We are doing presentations, half-day workshops, tabled events. So far in 2024, we reached over 6,000 people. In 2023, I call this the year of the state agencies. We were all over the place talking to regional offices for the DEC, Department of Transportation, Ag and Markets, and others. And this was really wonderful for us because not only could this allow us to reach a lot of people that were out there and at high risk for tick-borne diseases, but it allowed us to get the information out so that they then could transmit that information to other people. And again, going beyond what our small group can do and getting more people talking about the information that we're providing. Now, if you go to one of our in-person events, you're probably going to end up with a tick removal kit. These are very, very popular giveaways. Since the original kits were put together in 2018, we have dropped the mirror. Feedback was telling us people weren't really using them. They were too small for the purpose. And we have added small Ziploc baggies. If somebody were to remove the tick, they can then put it into the baggie and then have it tracked so that they can do either the ID themselves by using the tick ID card or taking it someplace to a county office. To date, we've ordered almost 10,000 of these kits. We purchased the components separately and we put them together in-house. We have discussed monetizing them, but so far we've been able to find room in our budget to continue to purchase the components and then give them away for free. These kits cost about $5 each, and we took that opportunity to gather data in exchange for the tick kit. People would provide us with information with a very short survey. And between 2018 and 2022, we collected about 3,000 surveys. Now, some of those are results. One of the questions we asked was, what avoidance techniques have you used? And the purpose of this was to give us the information for what should we actually be focusing on. So a lot of people are using repellents, so we probably don't need to touch on that so much, but less people are using permethrin treated clothing. So this could be something we would more heavily want to emphasize going into the future. We also asked more of an efficacy question. Do you plan to change your tick avoidance or tick removal behavior as a result of what you learned? And 84% of people said yes, they were absolutely going to do that. Now, as mentioned, I am getting my master's in science communication, and I took this project on as a class project, and I wanted to share some of those results for you. First of all, I did a literature review, and some of the information that I collected was that, despite connections to academic information on tick personal protection, consistent use of these measures is low. Increased worry can increase tick protective behaviors. Fear can reduce intention to protect oneself. And how people are exposed to information can impact their worry, anxiety, and fear. So this idea of emotions, especially fear, was really interesting to me. Increasing worry without pushing it into fear is a fine tightrope to walk. Our presentations are set up to start with the biology of tick and the risk of tick-borne diseases, and then we switch gears to focus on protection strategies and to build confidence. And we even have a transition slide for this, meant to use humor to kind of soften those emotions. I really like the term healthy paranoia because we want people to have ticks tucked in that corner back part of their mind whenever they go outside. But we also definitely want them to go outside because there are poor health consequences of staying indoors all the time. So to this end, I created a survey that was sent out to people that had sent back that earlier survey and provided their contact information. I got 426 responses, or 23%. I'm pretty happy with that, especially since the survey was a whopping 32 questions. Kind of pushed back on my professor with that, and nope, she thought all that information was needed. So those included questions about demographics, where individuals learned about the Get Ticked New York campaign, how often they use tick personal protections, questions to determine their emotions, where they get their tick information, and then I analyzed this. And in an effort, believe it or not, to keep the survey instrument at a reasonable length, knowledge gained was very superficially measured. So when we look at the frequency of recommended tick personal protection, for most of these, people are not doing them more than they are doing them. Some of the exceptions were staying on trails, conducting daily tick checks, and treating pets. So some good things, but some work to do as well. I also compared if they had learned something in the campaign to these different tick management strategies, and there was some weak positive correlations in terms of gaining confidence in their ability to protect themselves, tucking their pants into the socks and heating clothes and dryer. And there was a weak negative correlation that people that had learned something through our campaign were less likely to put petroleum on a tick to remove it. I wish that these were stronger, but we'll take what we can get. Looking at confidence, we also saw some weak correlations with the more confidence they felt, the more likely they were to stay on trails, the more confident to tuck their pants into their socks, and also to apply repellent to bare skin. The strongest correlation was with confidence and how many people they shared the tick information with. So this was really exciting because we're trying to get people to share this information. So people that were very confident were likely to spend it, like 90 people said that they shared it with 10 or more people. So that's 900 people that we weren't able to reach personally, that we were able to reach with our information. So conclusions, similar to other tick education programs, this campaign has not been able to demonstrate strong evidence of tick protection uptake. Weak positive correlations do show that we are moving in the right direction. And I think that rather than broad educational approaches for these ticks and tick protections, maybe smaller focused projects aiming at one strategy, like permethrin-treated clothing, for example, might be more successful. My capstone project is trying to determine the acceptance of these various tick management practices and to determine the degree to which risk perceptions and confidence constrain uptake. My next eventual step is to take that information, try some messaging, and then do message testing. We did get some information that I really wanted to share with you. One of my favorite quotes that I ever had from one of my presentations was, "That was terrifyingly wonderful." And here's a quote when somebody was responding to the survey: "So helpful and useful in reducing our concerns. Can't thank you enough for turning my anxiety about ticks into intentional and proactive actions that help keep our family safe." Thank you for your attention. I really want to thank my incredible co-workers, the teamwork to put all of this together, as well as the funding that we received from New York State, as well as the Extension Implementation Program grant. Thank you.
Thank you, Joellen, for a great presentation. I want to take a couple minutes and see if we have some questions here. And if everyone could please fill out the poll question as well, that would be great. So one question I see is, so many great contacts. Have you been able to see a decline in tick-borne disease or tick bite reports from pre-program?
I wish I had the answer to that. Those are both things that are beyond the scope of what we're able to collect. So, no, and if anybody has any ideas for how we could do that, we'll give it a go. But at this point, we haven't been able to do it.
Thank you. Joellen, did you want to put like your email in the chat or anything like that?
Oh, sure.
Bob says, outstanding presentation. Thank you. So Joellen's email is.
Yes, I am looking forward to getting my graduate degree done and then having more time to work on this other really fun stuff and take this all to the next level.
It's great. There is someone commented earlier that the claymation video is excellent, I think.
We love the claymation video. I don't know if Max is doing them anymore. I had linked to his YouTube channel. If that's something that you want, you can reach out to him, see if he's willing to do one for your project.
Another question, the tick card with the feel was interesting. Are those available for purchase?
So they had donated 120 for me so that we can put one in each of those education boxes. I don't know if they still have them or not, because that was back in 2018. It was Brigham and we'll look that up.
Thanks.
Put that in the chat. Brigham and Women's Hospital.
So check that out, see if they have it available. Do we have maybe one more?
And I don't know how hard it is to get those done. We haven't looked at it because we have the cards. I'm sure somebody can do it.
Well, it doesn't look like we have any more questions right now. So I want to thank you for a great presentation and for providing all of those resources. I think it's a great way to end the day and end the Tick Academy here. I want to thank everyone who participated throughout yesterday and today. And I also want to thank our sponsors again. I'm going to share my screen so you can see some more information about them. Start it over. So I think you can see all of them on the screen now. But we want to thank our gold sponsors, Tickbox Technology Corporation, Project Lyme, US Biologic, and Mainly Ticks. Our silver sponsor, North Shore Mosquito Abatement District, and our bronze sponsors, Alexis Chesney, MS and DLAC, and Central Life Sciences. We also want to take a moment and thank NCIPM, North Central IPM Center, for providing the primary funding for the TIC IPM working group. This work is supported by the USDA National Institute of Food and Agriculture, Crop Protection, Pest Management Program through the North Central IPM Center. And once again, we are currently in the process of applying for that funding again for next year's grant cycle. So if you participated today or yesterday or both days and enjoyed the presentations and want to support the work that we do, the best way you can do that is by joining the working group and attending webinars and considering writing a letter of support for our grant funding. So if you're interested in doing either of those things, please reach out to me. Josie just put my email in the chat. I'd always also love to hear if anyone has comments or opinions on how the TIC Academy went this year. We'll send out a formal survey in the near future here for people who participated. But there's also one more question. Will PowerPoint presentations be emailed? I do not have most of the presentations to distribute, so you're welcome to reach out to individual speakers if you are interested in the content of the presentation. But we will, both days of the TIC Academy were recorded, and we will be sending out those recordings to everyone who was registered, so you will have access to those at a later date that way. So again, I want to thank you all very much for being here. It was really exciting to see the turnout we had, and we're really happy that we were able to put this on for a 5th time. If anyone has any questions or comments, please feel free to email me, and we hope to see you at our monthly webinars and back here again next year. So thank you very much.